Hello. Okay. Pleasant morning to all. I'm Aisha from the Department of Biochemistry, first year MSc. Today I would like to welcome our speaker, Dr. Nagatihalli, and Dr. Chandrasekhar Joshi. And I also welcome all the participants and the technical team. Enjoy your day. Thank you.
Good morning, everyone. I hope you had a great 2 days of session. Hope you learned um, many technologies, research, uh, understanding cancer biology. I think today is a little bit longer day. Looks like we have more. I'll be talking in the first session, and then second session, Joshi, and then after lunch we into the tutorials like yesterday and day before yesterday. Um, so today, day 3. So after talking to all of you, I also discussing with you. I think I realized that I think more of PhD students here, right? And those who want to do PhD, and also those who are in MSc want to do PhD. So I thought giving you a little bit of story about the PhD program, right? Um, One of the story I selected, changing a little bit of some of the slides, even though the targeting cellular signaling and tumor microenvironment is the same, but the story I'm going to start with one of the student. Her name is Samara Singh. She did PhD with us, and she's now in the postdoctoral fellow in Johns Hopkins University. It's one of the great university. And I'm going to tell you her story when she came to do PhD, and what did she do, how did she conduct research, and you will get a story from her. And at least she published a great paper. Cancer research paper is not easy. As a graduate student, means the PhD student, publishing in a cancer research is a great paper. And also the suffer, and then the, how much patience you have to have as a PhD student, and also guidance, mentoring. The same time, learning, the passion you have to do the PhD. I think that is a story that you're going to learn today. You're going to see what exactly happened to her, and how did she finish, and how did she accomplish, and now she's doing even great. So I think it's important for you to know how to conduct PhD and what you go through. Probably she went through the same story. Okay, it's her story. Okay, we all know now therapeutic resistance, right? So when she came to laboratories, she started working on what projects we have in the laboratory. Overall working what projects, right? She started with that, talking to people, talking to MDs we have in our laboratory. We have a lot of medical students, those who have done MBBS in their medical school, and then they'll be doing the MD program, and all they come to a lab to do research. Any clinicians there, they do research 2 years. Most of them. If you want to get into a fellowship and go into a program like that, in a good university, academic environment, if you want to go work in a hospital, you have to conduct a research. Okay, 2 years of research is a great thing, right? That concept is good because even the medical students and the doctor, they will know how to do the research, and also they will know how to read papers, and if anybody has published any research, then they will know how to do these kind of things and what exactly happens when you conduct some research based on whatever they are treating with, diseases or whatever. And that's why they need to know the research base also. That's why they come to the laboratory. So she, as a PhD student, right, she talked to all these doctors, talked to all these, and graduate students. Already they are in the second year, third year, fourth year. Also postdocs, they're all done PhD. And there are postdocs, MDs also in the laboratory. And they will be doing the research in the different programs. So she will talk to all of them, and then she'll like figuring it out, what do I do, right? So mentor is not going to guide you everything. You have to come up with the logics. As I said, when you decide to do PhD, that means you're already scientist. You have to start thinking like a scientist. So you have to discover what you want to do as a PhD student. Though she started with, we had a therapeutic resistance in pancreatic cancer is a big, huge problem, right? You all know now. And remember, this is pancreatic cancer again. It can be any cancer. The story is the same thing. It doesn't matter which cancer how you want to do, what you want to do, but the story is going to be the same thing. So she understood that it's, the 90% of these cases, they have a KRAS mutation. KRAS is an initiator in the pancreatic cancer progression, and also it activates pro-inflammatory, networks. And she understood she read a lot of papers, and we had everyday communication, and we used to talk, and she learned all this, and also she learned signaling pathways, right? And you take one single cell from the tumor microenvironment, and if you enlarge and see, and you'll see a lot of signaling pathways. I showed you yesterday, like cell-cell communication, right? You saw the receptors and signaling between the cell communication, what exactly how they communicate each other, and what activates and all those things, right? So it's the same way, she went and searched all these pathways, and then she selected some of these pathways, and at the same time, what she was actually studying was all this STAT3 that we have done before. STAT3 is an inflammatory activator, and she also saw that STAT3 can activate all these IL-1 alpha, beta, IL-6, all this pro-inflammatory ligand. She studied all these things, but remember, you all start with something similarly. But you don't know where to start with, right? You know all these things, but where to start? How do you start, right? So next she continued understanding the therapeutic resistance. And then she took all this single cell RNA sequencing data, like I told you, public data system. Like she went to public data and she downloaded all these things, and she learned R. She learned R means it's a program. So R is a program. She learned R so that she can actually figure it out herself by downloading all this public data, and then she analyzing herself, making sure that, because going to the bioinformatician every day, every other day, and getting the data done, probably it's a big problem. But rather, all these postdocs, what they do, or the graduate students, they learn themselves. It's not a big deal. It's actually, you can learn faster. Like within a week, I think you can actually program it such a way that you can learn. It's not a big deal. You can actually learn. Anybody can learn anything. So she learned the program, and she started looking into different, cellular components and in the non-tumor and tumor, and also in the tumor, different components in the different KPC mice. That is a KRAS P53 mice she took, and the PKT mice that we had in the laboratory, another genetic model. So she took all this data and she was analyzing it. And also she knew that the stroma is a big problem in pancreatic cancer. She knew CAFs, and she also studied ECM, that is an extracellular complex, and she kind of went into a tumor microenvironment, studied all those things. She got all the knowledge of what exactly is going on in pancreatic cancer. What is important in pancreatic cancer? This is important. You get all this knowledge, and then she was searching our papers. What did we publish? Because when you go to a laboratory, one investigator laboratory, you have to search what that mentor or anybody working in that area or group, what they have published before, because it's important so that they will give you knowledge and the guidance. That's what she did. She went back and saw all these papers that we have published before, I told you this story, MEK and STAT3 story, inhibiting both the signaling pathway, what happened and all. Remember again, I'm linking our own studies, that's how they do. They link and study because the knowledge has been developed already. It's important to someone else to come and add that to the knowledge, some more information and get it better, right? So that's what she did. She took all this information, she read all the papers that we published, and she came and asked a lot of questions, right? And also, when we did this study, like this is a in vivo, that is a tumors they have taken, treated with either the STAT3 inhibitor or the MEK inhibitor, or together, and she took all this information, right? And also, at that time, there were a lot of papers published understanding the calf heterogeneity, right? The papers started coming. Calf is becoming like hot at that time, like 3, 4, 5 years ago, and now she understood that there are papers coming out as a calves, and you have to do the same way. Any PhD students understand, and at the same time look what papers are coming out, the one that you're reading, one that you think that it's important, that area, right? So she also understood that IL-1 alpha is a critical mediator involved in the inflammatory calf polarization. It is published data though. It's a published data, it is not ours or anything. She understood that IL-1 alpha is important mediator in the inflammatory calf. So she thought, okay, calf is important because this is all stroma in the pancreatic cancer, and now inflammatory calves polarization will be done by this IL-1 alpha. Now she got IL-1 alpha is important. She came and asked me, oh, Nag, this is what happened. IL-1 alpha looks like a key mediator, and looks like I'm interested in. You know what? What I did, I went back to my old data, and I digged all the data, and this was one study that I did long ago. That what I did was I took all this MEK STAT3 inhibitor, treated the samples, like these are all MEK STAT3 inhibited one, and MEK inhibited and STAT3 inhibited. These are the control. These are all cytokines. Okay, this is the cytokine array screening. So that's what I did long ago. When I analyzed at that time, and the IL-1 alpha and the IL-1 beta were very important because when you combined inhibition of STAT3 and the MEK, IL-1 alpha and the beta, they are here. When compared to IL-1 alpha and beta here, control, and if you compare any of the STAT3 and the MEK inhibitor, and you see here, IL-1 alpha went down completely. Means that when you inhibit STAT3 MEK together, IL-1 alpha and IL-1 beta were going down. Means that whatever the data we had on the tumor microenvironment, affecting tumor microenvironment, anything like that, probably mediated by IL-1 alpha and beta. That's what I told her. Okay, this is amazing. This is people are started looking into IL-1 alpha. Probably our data clearly shows IL-1 alpha and beta are important because of the data what we had, right? And then she thought, okay, looks like that is it. I'm gonna work on IL-1 alpha or the beta, right? Then she started looking into IL-1 alpha, right? So you have to start with something. So that gives you a confidence. If someone has published something and now you're linking your own study, and now you know that confidently you can move forward and say, okay, probably this is something that I can look into. Okay, that's what she did. She went and searched for all those things. And also what she did was she compared the IL-1 alpha expression in the normal tumor and to the tumor, pancreatic tumor. And she saw IL-1 alpha was very high in the tumor when compared to the normal. And then she took this PTF1A Cree and the PKT. This is the genetic mouse model. This genetic mouse model also she saw IL-1 was very high. She got more confident now. Now, okay, she thought at least I have aim 1. Remember, in your thesis, you have to have at least aim 2 or 3, right? So at least she thought, okay, I can start with aim 1, right? But she's not there yet though. But she has to figure it out. Okay, IL-1 alpha is high. Probably people might have published already IL-1 alpha is high. But important is that what is the importance of IL-1 alpha after that, right? I mean, in your study, in your story. So now she got, it is overexpressed in pancreatic cancer, and also she saw if you have a high IL-1 alpha, there is a worse survival in the patient data. These are all TCGA data, okay? Now remember, what she did, she collected the data of IL-1 alpha, she went to TCGA database, and she looked at the IL-1 alpha story, anything that data are available, and she saw IL-1 alpha, if you have high, that is worth survival. Okay, great. Now she got the story. Now she was thinking, okay, now I'm going to start looking into IL-1 alpha and see what other other cytokines can link together, and whatever that is. Okay, she can get information about. Any information you get is information for you to put it in your figure 1 or figure 2 in the thesis, right? So she started with that, and she started looking into, okay, now I got the story of IL-1 alpha. What about IL-1 beta? So people ask you, if you say IL-1 alpha, what about beta? What about something gamma? What about other? What is important in the IL-1 story? That what other IL-1 cofactors are there, or regulators? And then she starts searching about IL-1 alpha and IL-1 beta. Okay, we told no, you cannot just say IL-1 alpha is important. You have to compare with other marker, right? Like IL-1 beta. Then I'll ask, because IL-1 beta story is already there in the research publication. Then I will ask, okay, what about IL-1 beta? Did you look at it, right? So that's why she compared IL-1 alpha versus IL-1 beta. So both are pro-inflammatory, both share the same receptor, and they have a separate regulation. Okay, and initially what she did was, so she collected all these cell lines, and also human and also mouse cell lines. She separated these cell lines with a high IL-1 alpha and low IL-1 alpha, and high IL-1 beta and low IL-1 beta. And even when she saw that, there are many cell lines that she saw very high IL-1 alpha when compared to IL-1 beta. So IL-1 beta was less when compared to IL-1 alpha. So she got story. Now IL-1 alpha, if it is very high, that is important. I mean, looks like it is important. So her story is building, right? Now she got IL-1 alpha. Now we see IL-1 alpha is increased. So that's how you connect your story. You study, and probably she might have changed to IL-1 beta if IL-1 beta was high. Then she would have looked into IL-1 beta and whatever happens to IL-1 beta. But your story also will be important, how you are building your story. Even though you start with IL-1 alpha, probably you end with IL-1 beta. It depends on what data you are collecting, right? So then now she got to know IL-1 alpha is important. People ask her, okay, then how do you know IL-1 alpha, how it is regulating? So we all know IL-1 receptor is there, and it has to go through the receptor and show me the receptor expression and show me how this is IL-1 alpha is regulating through IL-1 receptor. So before doing that, what you need to know is that whether the receptor express or not, whether the receptor is there. Don't predict. This is science and the scientific investigation you are doing. There is no prediction. You have to have the data. You don't have the data, you cannot predict. You cannot predict and say it is actually happening. No, it may not be happening. Until if you don't have IL-1 alpha receptor, how is this IL-1 alpha is even working, right? That is important. So you need to know that how this is regulating, through what. You need to see it. So this is exactly what she did. We told her, no, you had to go and do the single cell RNA sequencing data and see whether IL-1 receptor is expressed anywhere in any of the cells. And what she saw, look at that, calves. So calves expressed high IL-1 alpha, IL-1 receptor when compared to any of the cell types. What does that mean? That means that the IL-1 receptor is high in calves when compared to any of the cells. What does that mean? That means IL-1 alpha is not on the epithelial cells and working. IL-1 alpha is going somewhere on the calves and hitting the calves because the receptor present on the calves, right? She got that idea. Okay, this is the data. Your own eye, you are actually seeing it. IL-1 receptor is present in calves, right? That's what she did. See, that is not enough, right? So, when you are doing the research, okay, now receptor is present. Okay, now you have to prove it because it's science. You have to prove again and again in multiple ways that same thing, right? Just not one time, just not one experiment. You have to prove in multiple experiment the same result, it is happening. That is the confidence you have to build. Confidence you have to build with the data, right? That's what she did. She did a lot of confocal microscopy. She did a lot of flow analysis just to show with the EpCAM positive, means that epithelial cells separated, the CAF cells separated. She just separated all those things. Okay, now looked at EpCAM positive, EpCAM negative, and the CAF positive, CAF negative, where the IL-1 receptor is present or not. She confirmed. She confirmed that IL-1 alpha is released from the pancreatic cancer tumor, and now it is going and binding on the CAFs on the receptor. IL-1 or receptor is there, and now it is going and binding, and something it is doing on the inflammatory CAF activation, or something it's doing. See, it's a story, right? Okay, now she got to know, okay, something is happening, my story is built in. Okay, now she's actually having the story. Now, next question comes, okay, she comes, okay, she's happy. Now I'll say, what mediates the tumor IL-1 alpha production? Now a question, okay, now she got a story, she's excited, now I'm depressing her, telling her that, okay, this is great, this is awesome, you got that story, you got all these figures, fantastic. Now tell me how this IL-1 is coming, how this IL-1 even alpha is produced. You need to know that too. Before you go and tell the story to me, that's what I'm saying. You can go ahead and work on it, but I'll ask the question, how this happened, right? So one story that we built it before, STAT3 and the MEK inhibition together, and the IL-1 alpha was regulated. IL-1 beta was regulated. So now she knew that data, okay? We already published. She can link easily to the MAP kinase and the STAT3 story. But she went ahead and she wanted to do her own investigation. We thought, wow, that's awesome. You never know. IL-1 alpha is, for example, IL-1 alpha is not just regulated by STAT3 and MEK, but is it regulated by many other signaling pathways. It's not one. In cellular signaling study, it's not just one signaling. Only you're studying one signaling. As I told you in the beginning, when the first day one, that there may be multiple signaling pathways affecting the same thing, right? So it's the same story, but she wants to do her own screening. She wants to study, understand how that IL-1 alpha is really regulated. Probably there may be other signaling pathway, maybe stronger than STAT3 MEK, okay, maybe weaker than STAT3 MEK. At least she has a story. If it is weaker than STAT3 MEK signaling, and she can tell that STAT3 MEK is a stronger, and when compared to many other signaling, she has a story. And if it is other story comes in, and she can say, when compared to STAT3 MEK signaling, regulating IL-1, I found a new signaling pathway which is more important than STAT3 MEK, like that. Okay, you have this, either you can make the story, and you have that, you can make the story too, but in a different way. But you have to find a story, right? So that's what she did. She went and looked at the TCGA database again, okay? And what she did, she separated the IL-1 alpha high, IL-1 alpha low patients, okay? It's a data, you don't have to do anything. You separate them, if you know how to do it, because you have bioinformatics analysis, and separated those, and she saw all the differentially expressed gene expression, and she did conducted analysis. So what she found, that one of the signaling pathways, she saw MAP kinase was high in IL-1 alpha high patients. So she came back to the same signaling pathway, MAP kinase, which we studied, right? And that's the story. She's actually linking back. Right now she got the story, MAP kinase is important because of the expression is very high in the IL-1 alpha high patients. Now she was happy. She was very happy. She came back and she said, MAP kinase is good. There are many other MAP kinases. MAP kinase 1, 2, or P38 MAP kinase. And at that time, MAP kinase ERK was very well known. She wanted to check P38. And I asked why you want to try the P38, because there were papers coming out, not well established signaling pathway, even though it regulates like a MAP kinase, but there were papers, very few papers were published in that angle and area, and she wants to establish something, whether P38 regulates the IL-1 alpha or not. So what she did, she compared all of this signaling pathway, okay? She just did not go and selected the P38. Remember, she has to prove P38 is important. But even though she thought about it, but you have to prove scientifically comparing with other signaling pathways, right? What she did, she collected the inhibitors for mTOR, MEK-KERK, P38 and also Jun kinase. These are all come in the same signaling, KRAS signaling pathways. So she selected all the signaling inhibitors. And then she selected all these inhibitors. She started inhibiting each one. Okay? So inhibiting each one and see which one, when you inhibit, IL-1 alpha will get downregulated. Right? So then she will select that, whichever that is coming out. So close her eyes, she got all the inhibitors, and one day she started working on, okay, story, right? And she came back with this one. She's like, she got excited. The reason is the IL-1 alpha went down with the P38 inhibitor when compared to any of the other inhibitors, even the MEK inhibition. Okay, even though there is a little bit of change here and there, but the strongest inhibition she got is P38. Okay, this is exactly you have to do. What you have to do is, it's not that you pick one and go. No, you have to compare. Make sure the one that you're working on is the strongest, is the pioneer, is the one that actually regulating this signaling pathway which you are thinking of. You know, basically she is linking the story. Right? It is not that if she would have got mTOR, she would have started with mTOR. If she would have got the JNK, she would have started with JNK. It is not that she wanted to do only P38. It is just that. So it's about what data you get. You have to go with the data, not with your prediction. Right? You can predict, but end of the day, your data has to say that. Right? So that's what she did. So then she got excited. She got another story to talk about maybe AIM2 or AIM1B or AIM1A in a thesis. So she started with, okay, MP38, she got, okay, freshen up and started with MP38, she got inhibitor, and now next question come, okay, what you gonna do, right? Like start looking at the picture, she already started with the literature, P38, what is happening in other cell line, other cancer or other diseases, where is this P38, how this is regulated, what activates. She was like, one day she was happy, another day she was suppressed and depressed, and all these things happen. But you know what? It's a process. It's a process. You study papers and you get a lot of knowledge, and that time you have a lot of questions, right? But the questions, you cannot answer all the questions, but you have to answer the question that which you are working on. And what she did was she started digging how this P38 is regulated, what it regulates. Okay, she came and asked me, okay, whether P38 has any role on anything regulations. Then she saw CREB here. She saw CREB. Oh, you know what? P38 activates other transcription factor. It also activates CREB. That's yours work. Okay, we were working on the CREB. I told you the story in my first talk, and she thought, no, look like our story is linking. Okay, great, linking. I don't know whether P38 regulates CREB or not. There are papers published, but it's okay if it is good. Or the only thing is matters is that whether it is helping you to investigate. But we can actually take the story later on. Again, I'm telling you, they all linked in the story in the later. But important is that she has to graduate, right? At the same time, she has to publish a good impact paper. Otherwise, she will not get graduated. And also, her story has to be true and novel, right? She has to build a story which is actually novel, true, and it is happening, right? It should, so she should not take the CREB and kind of dilute your work. You focus on what exactly you're trying to investigate, right? So that's what she did. She forgot about CREB and she forgot about everything. Just focus on P38 and focus. You can easily go and start with something which is maybe someone else is doing neighbor, someone else, maybe other investigator, another university. It is not important. Important is that what you're thinking, the focus on whatever the data you're collecting and getting it, focus on that particular one, because that may be the true thing. Rather than someone else may be doing something and you start working on that and see whether that works for you. It may not be true, okay? So that's what she did. She went back and started working on P38. She looked at all the cell line again on the Western blot and looking at what cell line express high, what cell line express low, so that she can actually model those cell lines to study further. And she did all those analysis, and also she did IL-1 alpha, expression in all these cell lines, and she compared which cell lines. Like for example, if you take SW1990, okay, or the other cell lines which are high in P38, also high in IL-1 alpha. So she selected. It is not that she only did these ones. Remember, she might have done multiple other cell lines. She screened all the cell lines. She selected the best out of it. She selected the one which actually have a high P38, high IL-1 alpha. That is her story, right? When you have IL-1 alpha high, P38 was high, and vice versa. So she wanted to select in the patient data. So she selected those cell lines so that she can play with it in the in vitro experiment, or you can actually play with it in the in vivo experiment to prove our hypothesis and logics. Okay, she correlated all these things, making sure that they all correlate well. Okay, and then she went ahead and did the correlation between different human PDAC cells. Like she took some of these MAP kinase and the SW1990, and then she did the siRNA for the MAP kinase, making sure that when she did the decrease in the P38, what happens to IL-1, and also she did the low IL-1 alpha, IL-1 alpha story. You can see the MAP kinase expressed low IL-1 alpha, and also she saw SW1990 high IL-1 alpha. Remember, picture perfect. I always say, even though she had data with the Western blot and other analysis, ELISA and all those things, but picture, you have to see from your eye. That's what I said. You have to show me the picture. Show me the picture which actually I can see IL-1 alpha is there, right next to P38 or together with P38, somewhere around, right? You see that there. She saw, okay, now I got the data. Look at that. It's actually co-regulated. They are actually right there. And the red is at the P38, and the green is IL-1 alpha. And she saw beautifully that they both express, and she was very happy. Remember, important is that she selected the best based on many factors. When you are working on like that, you will never go wrong on the way, unless it is contaminated, unless there is something else is happening to your cells. But you'll never go wrong. It is there, it will be there forever. Okay? The data reproducibility only in science can happen, only the data you collect like this. Otherwise, you'll never get the reproducibility of your data. If someone else is doing it, they may not get it. Okay, it is important you take this data and conduct in experiment SW1990. It will express high P38 when compared to MEPACA. It will express high IL-1 alpha. Doesn't matter who will do it in whole world, right? That is, I think, the important thing in scientific world, right? And what she did, now she got even more excited. She got a model. She got these cell lines. Now she can model a lot of things, right? But remember, that's not enough for her. What she did, she went back and immediately, we had all these model in our laboratory, like human PDAC tissues and mouse models and all those things, right? She wants to go ahead and look at in all these things before she do any experiment. That's what we say. Okay, just don't get excited. Go back and look at all these things, making sure that your work, because you're going to spend next 2-3 years in the laboratory, or maybe 4 years, like working on these models. Make sure that work that today you're showing me is perfect. Is going to be reproducible. You will not have any problem in the future. So otherwise, you have to change your science, your aim, which is actually even more worse than just doing the science and working in the laboratory, right? So that's why when in the initial stage, you have to make sure your data, what you're collecting, is perfect and reproducible and repeatable. And anybody you can give it to, they should be able to get the data same way, right? These are all the things I think she has to think about. And what she did, she went back and got the PC3017, that is a PDX. We have these PDX models in the laboratory, the patient-derived xenografts, and we have collected our own patient xenografts, so we know the mutation profile of all these patient-derived xenografts. So she selected those, and she saw that one of the tissue that they got high P38, have a high IL-1 alpha, something like that. And she also saw all these patients whether P38 alpha, which is high in a tumor when compared to normal. And then she went ahead and looked at many other human tissues, and she wants to make sure they both co-regulate. Means that P38 and also high P38, low P38 in a patient tissue, so that she wants to make sure that she collect all this data before she move forward. And also she went ahead and looked at the different mouse models we have in the laboratory, like KC. KC means only KRAS activation, and the KPC is a KRAS activation, and also P53 mutation. And another model we have in the laboratory where KRAS mutation and also TJ beta receptor knockout model. So she collected all these tissues from other investigators. Remember, this is a collaboration between the colleagues. If you see in our paper that published, so there are many other investigators in the laboratory that they are working on, PhD students and other. So they all talk each other during lunchtime, and they collect all the information. Hey, can you give me your tissue? I will also look at IL-1 alpha. Okay, yeah, but you have to give me this. You have to give me that. So it's like that. It's a give and take business. So there also there will be a fight. There is other things, right? But make sure that your colleagues, you're friendly, and you're having a nice dinner, lunch with them, and having coffee all the time, because they are the one giving you data. Okay, they are the one giving you a material, so you have to be good with them. So that exactly, so she collected all this tissue from different people, and then she looked at P38 and IL-1 alpha, and she got all the data. Now she saw when compared to KC here, like right here, when compared to KC, only KRAS mutation in the cancer, you all know already, but only KRAS what happens. I already told you the story. But in the cancer, when compared to KC, is high. She saw high IL-1 alpha, high P38. So she's basically getting the data exactly what she is thinking about. You may say, oh, like this so perfect. Yeah, it's perfect because the model she screened, the data she collected in the basic initially is actually going well with her data later on. So she might have tried in many ways. Like for example, the major issue here is that probably the antibody did not work, maybe did not express, maybe there is an issue with the antibody that she got from the one company. These are all the small technical issues will be there. But important is that figuring it out, all those things, and coming here in the end, perfect data, right? Like that. So there's a correlation study. So now she is ready. So when you want to make a AIM-1, you have to have the background story. She built all the background story. She is very strong now. She got a story, P38, IL-1 alpha. She's ready to rock, right? So now she got the AIM-1. I'm so happy that I got a AIM-1. Okay, what is the AIM-1? Determine the mechanism of P38 MAP kinase mediated transcriptional regulation of IL-1 alpha in PDAC. She got already all the data, but she wants to regulate and see what is the mechanism, how that is regulating. The P38, she got a story, IL-1, she got a story, but whether P38 is regulating IL-1 alpha, you have to show in the transcriptionally, right? You have to show further that whatever the regulation she is showing, she's actually showing the correlation, not the regulation, right? You have to show whether they regulate or not, whether they regulate or not. If they are not regulated, it's fine. There is always something else will be there to work on. If they are regulated, it's fine. I'm completing the AIM-1. So that's what she is actually working on. She worked on the next experiment where she wants to prove that P38 is regulating IL-1 alpha. So what she did initially, she did the P38 inhibition in different time point, different concentration and all, and she screened all these high IL-1 alpha cell line and low, and whatever that you guys saw the data. She selected the high IL-1 alpha cell line, low IL-1 alpha cell line, and the medium expressed IL-1 alpha cell lines, and she started inhibiting the P38 and looking at P38 inhibitor and look at whether the P38 is inhibited or not. Okay, these are high IL-1 alpha, low IL-1 alpha, but she's using the p38 inhibitor to make sure that whenever she goes to the next experiment, this is not going to haunt her again, something making sure. Okay, she analyzed all those things. She also analyzed the IL-1 alpha release from these cells. High IL-1 alpha, low IL-1 alpha release she saw. Remember the figure I showed you. The IL-1 alpha has to go to the IL-1 receptor on the calves and regulate in the calves because there is no receptor in any other cell types, right? That's why she has to look at the IL-1 alpha release from these cells, right? And also that's a key thing. That's why she did ELISA to look at the IL-1 alpha release, okay? Making sure these cell lines are cell lines and making sure these cell lines are really going to work for her. And then she went ahead and knocked down the P38 and see, because what she did all these things is all the drug study, right? Drug can be anything. It may be off target. It may not be directly regulating your cell thing. The reviewer is going to ask you, okay, did you do genetic knockout or did you do genetic knockdown? So anything that you can show in a drug may not work in the genetic knockout. Also, the vice versa is also true. You have to, if you are saying that it is P38 is regulating IL-1 or vice versa, whatever, you have to show genetically that they are the one. But you are saying is it's not the drug you are saying they are regulating. That means that genetically they have to regulate, right? So she went and did the knockout gene, and she saw that reduced IL-1 alpha expression in the P38 knockout. So it's basically she's seeing whatever she predicted on the drug study that it is happening. Remember, it may not happen all the time. Okay, it may be different, but drug can be off target. It can inhibit other signaling pathways too. Okay, so you have to make sure before you go, further you're doing anything that it is happening the way that you're thinking. So then she did the promoter assay. So now she wants to make sure P38, regulating IL-1 alpha, or at the transcription level, you have to look at the promoter. So if you don't know, you can actually ask your colleague how to do this analysis. There are softwares available now. Now you can look at easily promoter analysis and whether they regulate or not, easily available online. There are many softwares and the promoter and assays and all this. You need to collaborate with someone who actually knows all these things, and you actually can see whether your molecule is going to bind on any of these receptors or the IL-1 alpha or beta or whatever that is she was thinking. So she did some collaboration with someone, and then she got all the data with the SP1 and the RELA, because they are some of the regulators available. Andre means that it is already known in the field that there are this IL-1 alpha regulates SP1 and the P65. So she did all the literature survey. She collected all the information. So she put together and she collected, okay, marked here, and you see the color pencil everywhere on the paper, and she just took it out. One day you like cracking her head, okay, what transcription regulators in a promoter analysis I should take. There may be tons of them, right? Because each one will regulate tons of them, but you have to select the best out of it that actually works for you. It is not that SP1 and P65 she collected. She collected a lot of them, but these are the ones who worked, and she went ahead and published those data. But it is not that they are the only one. Okay, there may be other ones, but they did not work for her. It's about that. It's about screening but selecting, right? And she's going to tell you why she selected these promoters, transcription regulator from the P38 assay, IL-1 assay. So then she collected information on the SP1. SP1 is well known in the field. You all know SP1. Probably, if you are working in the biochemistry or biotechnology. SP1 is overexpressed in pancreatic cancer. She saw that. And she went, before she takes up any of the SP1, people ask, reviewers will ask, okay, if the IL-1 is expressed high, if IL-1 is regulating SP1, is that, IL-1 high as SP1 high too? Or whatever that P65 you are saying, if they are regulating, that means they are regulating genetically, means that they have to regulate, on the protein expression also. So she just saw SP1 is overexpressed in any of these mouse models she saw. She did see that, okay. And then when she started inhibiting SP1, and P65. Now, if the SP1 P65 is regulated, and if she is inhibiting these ones, I mean, they have to regulate IL-1 alpha transcription, right? And also, if they are regulating IL-1 alpha transcription, what does that mean? They have to regulate IL-1 alpha release, right? So this is basically, she is developing a mechanism and telling a story that this is how exactly this biology is working. Okay, she can go ahead and do the CAF analysis and all. It's fine. The reviewer is going to ask you, because you are building a story. You are a PhD student. If you are a PhD student, you have to analyze, understand the biology. You have to learn how to do all this experiment, how to conduct the promoter analysis. You have to see how they regulate each other. You know, there's a story you have to build. You can go ahead and do the therapeutic analysis, but it's easy. But again, you will not be learning. You'll become a therapeutic person, but you have to be a biologist. Means that you have to understand this biology inside what happens, right? So she did inhibit all this, and then she saw that IL-1 alpha is reduced, and also she did all this analysis. So basically, now she confirmed that P38 is regulating these two factors, and these are the ones regulating IL-1 alpha. So now she was clear on this, that the whole story is working for her. But remember, these two she selected the best out of best, and she making sure that P38 is regulating transcriptionally and also genetically, and these factors before she went ahead and did the IL-1 alpha study, right? So now she got a P38 story, but she's not happy yet. You know what I'm saying? So she's not happy because anybody will ask you, okay, P38, whether they are phosphorylating or not, whether they are phosphorylating, whether it's only transcription regulated. So she went and did the knockdown of MAP kinase, and whether she wanted to see whether they phosphorylate the SP1 P65 also in vitro. So she did see that the decrease in the phosphorylation when she knocked down the P38, and then she did all the different cell types. Again, I'm going to ask her if she is going to use the P38 inhibitor. I'm going to ask her, okay, if it is genetically it is true, can you show me with the inhibitor? Because you're going to use the inhibitor, and inhibitor, if you're using, and therapeutically if you're saying that P38 inhibitor you should use in the patient, means that I want to make sure that they are regulating all this SP1 and the P65 that you're talking about, right? That's what she did. She went back and did the chemical inhibitor, and then she saw that phosphorylation of these factors were also regulated. That means that I'm confident that now I can say that you should take this drug to the next in vivo work. But she was not. She wants to study further and make sure even further more. You know what I'm saying? She was more into a science because she got excited and she wants to do more and understand more before she go ahead and do the treatment, right? So what she did next is that she separated the nuclear and the cytosolic fractions, and she wants to make sure that in the nuclear, when she is inhibiting this, and the nuclear SP1 and the P65 are regulated or not. What is happening to the cytoplasmic, things like that. So what she saw was in the nuclear fraction showed reduced nuclear phosphorylation of this SP1 and P65. It's important to know, because you all say about the transcription regulators, if you don't show all these nuclear fractions and all, maybe probably it's doubtful whether they are really doing it or not, right? So even though it's very difficult to get the nuclear fractions and study, but she did her best, whatever she can, to make sure they are not like an opposite to what she's thinking, right? Before she do any in vivo assay. She was very confident now. She got all the data, whatever she wanted to do, and she showed all the data to us, like, okay, sir, this is all working perfectly. This is what do I do? Can I do it and do the in vivo? Yes, go ahead. We have a mouse model in the laboratory, breed them and get these mice and then start treating with the inhibitor that you're using in vitro. Okay, we want to make sure. You may ask me a lot of things like, did she do the proliferation? Did she do the colony formation? Did she do that one? She's done everything. Okay, only thing she's not showing all of them, but it's not important, because she's the science that she's working on, the science she's developing, the paper that she's looking into, but for that, what she needed, she has it. Okay, and the paper you target, and also important, as I told you yesterday, And her paper, she want to go higher journal and want to hit the bigger journal. She wants to make sure the mechanism is right, and she wants to make sure that she's going to do whatever the in vivo, she's conducting the same way. Similarly, I will ask her a lot of questions. Okay, whether this is, if this is happening and that is happening too, and how this is happening, how that is regulated. And can you show me that? Can you work on this, right? There is no frustration here. If you want to work and publish a bigger paper, there is no frustration. You have to go back and start working, right? That's PhD program. So she selected this, mice which we had, and she bred all these mice, and then she looked at whether the P38 inhibition reducing the phosphorylation of both SP1 and P65 before she says anything about the survival, anything about the tumor, because it can happen there, but what if this is not regulated? Then reviewer will ask, okay, can you show me in vivo whether this is happening? Right? That's why she selected this. And did she did that SP1 and also P65 expression in the tumor tissue. These are all the tumors. Like for example, n is equal to 6 means that 6 mice, pancreas tumors she selected, and the 5 of these, and she did all the screening, and she actually showing what it is, whatever it is there, right? So did she that decrease in the SP1 and the P65. So next, okay, now we know that P65 and SP1, bind, P38 inhibition reduces these ones, and whether this is anything happening to the IL-1, promoter and human PDAC cells. So what she did all these, chip qPCR analysis in a high IL-1 alpha producing human PDAC cell lines like SW1990. The P38 inhibition is significantly, reduced, binding both the P65 and the IL-1 SP1 to the IL-1 promoter, suggesting that both these factors regulate this IL-1 alpha transcription downstream of this P38. So the potentially whether the cooperativity of between the SP1 P65, there are a lot of regulation she did. She collected a lot of this assay, making sure the P38 inhibition with the pexmetin, she used that one of the drug that actually regulate the P65 and SP1. They are regulating the IL-1 alpha, making sure that this pathway is working in vivo. She wants to use it, making sure that they are working the way that she's thinking. So now the question comes is your question, right? You saw calves. You saw calves have IL-1 alpha receptor, IL-1 receptor high. So now she has to go there, making sure that that is actually working the way that she hypothesized. She did that inhibition. She went and did the single cell RNA sequencing. She collected all the tumor cells and the different EpCAM positive cells and the IL-1 alpha and whatever the expression values is, and trying to validate all this study in different cellular components. So she also did the tumor cell cluster analysis, making sure that all these signaling pathways, what she is thinking, each cellular components are cooperated and everything. And then we all knew that the therapeutic resistance, right? IL-1 alpha. I'm going back to the story, making sure that the inflammatory calf that I highlighted here, right? That is actually what I think she wants to look into. So making sure that IL-1 alpha, IL-1 receptor expressed in the stromal enriched population. So she then went and did all this, human and pancreatic tumors, whatever. She collected all this data. Remember, she also collected a lot of these single cell sequencing data sets from the human single cell sequencing and the mouse single cell sequencing data set, and she did all this analysis from the publicly available data sets, making sure that where is this IL-1 receptor, is it in the which population, before she did her own experiment, her own single cell, because she has to invest a lot of money, time, effort and collaborate with a lot of people and get those data. Before she wants to do that, making sure that it's available in the public data, whatever that they are showing, so that she can cite others' investigation, what the others have done in the field, right? So that's what she did. I don't have to go in detail, but she selected the cell types, fibroblast cell types, and she looked at IL-1 receptor, which is actually IL-1 receptor here. It actually expresses high in the fibroblast cells and also some in the endothelial cells. That is exactly what she is getting. So exactly what she thought about, we actually saw in the single cell sequencing data, and now she can see in our own model. And what she next did was understand the role of P38 in the MAP kinase in the inflammatory calf activation. That's our next question. So one, she proved all the mechanism. That is actually this one, how this actually regulated and all those. Next aim is that, as I said, she has to look into the calves and IL-1 alpha, how that is regulating in the through the IL-1 receptor, and that's the second story, right? That is the second story she is looking into. Now this IL-1 alpha, whatever it went up with the high in the P38, now this is going and binding on the receptor 1 and regulating inflammatory calf activation. And once it is activating here on the calves, remember there are a lot of other cytokines also get activated. Okay, so these are the cytokines she is actually mentioning here, because we all work on those cytokines. We worked on IL-6. I worked on the LIF. I showed you in the day 1, and CXL1 I did not show you. There is a cancer discovery paper we published, and it's a high impact, I think 38 or 39 impact factor paper. That is actually we worked on the 6CXL1 story. So that's what she's actually showing, that they are all getting regulated in this study. Okay, it's good. So basically she's linking all the story that what we published also. So she did with the pexmetinib, that is a P38 inhibitor now, and she is making sure what happens to whether this P38 inhibition has anything role on the IL-1 alpha mediated stromal activation. Is there any role on the P38 MAP kinase on the calf, extrinsic or intrinsic? She wants to make sure, even though literature says this, that, this, you have, as a researcher, you have to make sure that literature also is right, and you're also right. I keep saying this from the day 1. Doesn't matter what people have published, and when you are publishing it, show the same data again, it's fine. If that is true and it's true in your data, it's fine. You can show. No reviewer is going to say, oh yeah, you're repeating the same experiment. No, nobody says. But you repeat and then you show your data what is exactly happening. I think that's how you should link. That's how you should cite other papers, making sure that your data is collaborating, cooperating with other data too, before you show what is that new investigation you have done in your field, in your study. Okay, now she took the second aim. She started working on the P38 inhibition, whether it impairs the inflammatory calf polarization, right? If that is happening, so remember, all these experiments you have to do in vitro, in vivo, prove everything second time, third time differently, right? One may work, one may not work, right? But you have to prove it though, okay? If it is negative, it's fine. If some experiment is not working, it's fine. But you don't have to change it, okay? You just have to make sure if the experiment is not working, it is not going to work for anyone. So you don't have to even, you want to show that it is not working, it's fine. If you are not showing, because that's the reason that you have supplementary data, where you put all the negative data, also show them that this did not work. But, reviewers may ask, okay, why it did not work? That means it may work or not. Not all the reviewers going to ask, but you as a scientist, you will know how to answer if you are conducting this experiment correctly. Okay, you can tell them this is how I conducted experiment, this may not be working. I will be working in the future in the laboratory, we will investigate those. It's fine. We are all human beings, we understand it, right? That's how it is. So she basically went ahead and we did the P38 inhibition. What she saw was here, she's putting the P phosphostat 3 inhibition and all. Remember what we were talking about, the MAP kinase inhibition and then the STAT3 going up. Remember, in our case, it's opposite. P38 inhibitor is not doing that. It's actually doing the other way. But it's okay. It's an interesting story. I'm going to tell you a little bit more on this later on, why this is happening, why she's actually showing this. And in the summary of the AIM2, there's a AIM2A, that's what she just showed, the calf, just the interaction between the IL-1 alpha towards on the IL-1 receptor, right? She just got all this reduced inflammatory calf activation, and she saw all these IL-1, IL-6, LIF, CXL1, that we are all working on all those cytokine factors, chemokine factors. And in the AIM2B, what she asked herself, that can calf intrinsic P38 MAP kinase inhibition impair any inflammatory activation on the cancer associated fibroblasts, that is the CAFs. So she started working on the AIM2B here on that question, right? Now she is going away from the tumor intrinsic to tumor extrinsic, though in the tumor microenvironment. So exactly, our title was targeting tumor microenvironment. That is what I think now we are going. From the signaling pathway, we are moving forward to the tumor microenvironment. So, right, specifically. So, okay. So now the first question is whether the P38 inhibition impairs the inflammatory stroma. So what she did was, she did some of the stromal markers, and phosphostat3 was an inflammatory marker, well-known marker. She saw that inflammatory marker CAF also expressed STAT3, so she wanted to look into the STAT3 and also P38, and she did that. And she analyzed all these IL-6, CXL1, CXCL8, all these inflammatory CAF markers, cytokines and chemokines, and she did see that inhibition of all these major cytokine chemokines before she finalizing to go ahead and do the chemotherapy, whatever the study that you're thinking therapeutically. And blocking this P38, whether it reduces the IL-1 alpha mediated inflammatory stroma. She looked at all these markers again without the P38 inhibitor, and the IL-1 alpha activation means that she activated with IL-1 alpha and inhibited the P38. So she did the vice versa studies too. She is making sure that whatever she wants to do, and like Anakindra is another inhibitor for the IL-1 receptor. Okay, if she blocked the receptor, IL-1 receptor, now she is adding IL-1 alpha, it should not happen anything down because she is blocking IL-1 receptor. If you don't have the receptor, it will not do anything downstream in the CAF population. So that's exactly what she is doing, making sure that it's not happening, right? And then she did the MAP kinase inhibition, whether this reduces the fibroblast markers. She went and looked at the fibroblasts here in the single cell RNA sequencing she conducted, and now she's screening all the cytokines in the fibroblast subculture. That means, remember, these are all the single cell RNA sequencing. She got the population of each one, and she is looking into the transcription regulator in each population, making sure that these transcription regulators are all, cytokine chemokines are all regulated before she go ahead and tell you the story of the inhibition. So she saw all these were regulated, and the one that she showed, and also the one there are new factors here, and subcultures in the subfibroblast cells specifically. Okay. Then she took these fibroblast cells, and then she asked question, what else is going on in the whole population, just not the transcription regulators, there's not cytokines and chemokines, what other things are going on. And she looked at all these different signaling pathways. She has the collagen pathway, there's a TNF alpha, they're all important signaling pathway, making sure that you know all these things before you finalize your data, right? The pharmacological P38 inhibition reduces the fibrosis. She wants to make sure the fibrosis was down. That she used some of these markers for the fibrosis, and she did all those fibrosis analysis, like Tichrome blue and the Sirius red, all this standard you all know. So making sure that this fibrosis was reduced. Why she is doing fibrosis? So because we are talking about the CAFs. CAFs are fibrotic components. So she is making sure that the CAFs were regulated when she uses the P38 inhibitor. That means nothing is, the IL-1 alpha not coming, and it is not doing anything to the fibroblasts. That means that fibroblasts are down and maybe affected, right? That's what she is doing. Then she did the orthotopic model. Remember what model that I showed you? So one model is not enough. I keep saying you have to show it in a different way. Then I ask her, okay, you know what, this is genetic model, this is a very aggressive mouse model, what you used? And can you show me the progression model? In the patient, in the progression, cancer progression happens, a long time. Okay, the model that KPC is a KRAS and P53 mutation that takes a long time. This is like 12 months to make the tumor. In the PKT mice, it takes like how many weeks? 4 weeks. You get the tumor, 8 weeks they die. In the KPC model, it takes 12 months. It makes like 8 months, then there will be a tumor, and it takes forever. It makes the to get into a metastasis and a lot of other things. So she took this model. So she took the KPC model. is she injected orthotopically to the pancreas and make the tumor, and she wants to study, make sure in another model that the data that she collected is the same and true in another model too, and that's all. So she just repeated all the markers like I showed you in the other model. She repeated again in this model and making sure that they are all working the same way like I showed you before. So what she did was next CAF specifically. CAF specific deletion of P38 MAP kinase. Okay, whether our question was P38, whether they express on the CAF or not. Okay, that there was a single cell sequencing data she showed. The P38 was also expressing on the CAFs a little bit. Okay, just not in the tumor cells. She wants to make sure whether the P38, whatever the inhibitor she is using, whether it is from the tumor it is affecting, or in the CAFs maybe the P38 inhibitor and inhibiting it, that may be affecting. So just kind of like that, and she did all those, making sure that COL1A, there's a mouse model she developed with the MAP kinase knockout, and she just saw that Sirius red was low. That means that fibrosis was decreased. So when this is the CAF specific P38 inhibition, it actually suppresses the IL-1 mediated inflammatory polarization. She saw all those studies. That is actually in the AIM-2B. She's now very happy that her AIM-2B is completed. What is she missing? She's missing therapeutics, right? That is what I think we're interested in, as in a biologist in the transcription, translational studies. So she asked to do the therapeutics. That's what she did in AIM-3. All she did is making sure that we are all happy and we can take the study further, and making sure that she can publish in a journal if this works, if in vivo, and she has to show in a different models, and making sure her biology is fine, her biology already working fine. Now she actually showed all the regulation, cancer, how that is signaling pathways were regulated and everything. Now she's going for therapeutic and making sure the therapeutic works or not. So then she blocked the P38, and it actually showed the IL-1 alpha mediated inflammatory stromal and different experiment, just to make sure that before she do the in vivo experiment, the CAF specific P38 MAP kinase, and there is a loss in the intertumor MDSCs, and she just started looking at the different population in the tumor microenvironment, making sure that the P38 inhibition is affecting a lot of other markers, like they are immune markers, like ARG1. She is looking at the macrophage related markers, MDSC related markers, myeloid cells, and all those things. So making sure these are all working, and then she went ahead and did the T cells. The T cells are very important. Now if you see immunology, immune cells, T cells are one of the things, whether she should do the immunotherapy or not in this model. That's why she did the T cells, making sure that all this, she just saw the CD4, CD8 are all upregulated when she inhibited with the MAP kinase, right? These are all the models she's actually showing. She's just proving all the hypotheses before she go and do the therapeutic, okay? So, and also, there are a lot of monocytic populations she saw. She took this monocytic population from the single cell, and you can actually isolate your own cell type. So from these you can actually select these cells and do a lot of things. There are markers available now. You can actually specifically select those cell types, which one you want to study, and actually isolate them and study them transcriptionally and also protein level, right? And that's what she did. She wants to make sure whether this inhibition is affecting. Remember, like this, we never did in our SARC, EGFR, MAP kinase, all the story, because of the technology. At that time, there was no technology. We didn't know how to isolate all these things. And when we got the technology, then we are asking her to do all these things, isolation, making sure. If we had all this technology at that time, we would have done like the same way. But we didn't have the technology. So when the technology, you're getting the new technologies, you have to adopt those technologies in your study. So making sure that the worldwide, everyone reads your paper. Otherwise, they are not going to read. They are not going to cite your paper. It is right. Exactly. You have to compete with the world. Means that you have to compete with other investigator or whoever is actually conducting those experiments with the technology. You have to adopt those technology. If you cannot adopt or conduct experiment in your laboratory, you have to download all this transcription, regulation, and all these factors from the public database and show them correlation of something, so that it will actually relate to someone. At least they will be happy. At least you tried your best, whatever the facility you had, right? You had to try though. You cannot stop it. Okay, that's what I'm trying to tell you. So she had all this, technology. So she was able to show all these markers from different population. She's selecting different populations. She's showing that IL-1 beta also happens. Remember, what IL-1 beta she was saying is not epithelial marker. It is actually inflammatory, mediated. So that's what she's trying to tell you, that monocytes and the macrophages have the IL-1 beta. And when you inhibit with this, whatever the P38 inhibitor, she's now started showing IL-1 beta. Then I ask, okay, we said that IL-1 beta was from the inflammatory markers or from the other cell types, and if that is true, either P38 is regulating IL-1 beta, right? That question will come. Reviewers can ask you, right? So she basically just showing you that it is P38 inhibition is also affecting the IL-1 beta, which actually she started looking at, right? She won the mechanism and everything she did, but end of the day, that question came to her that she asked to look at the IL-1 beta, and well, she did inhibit. You can actually see that. Maybe that's not great, but actually it did affect the way that she was expecting. So also she went and did the macrophage population. Remember, she's actually taking each population and studying, making sure that it is happening the same way. It's a complete story. Then only you can actually get this paper to high impact, right? It's actually, you have to develop the story such a way that making sure that you're not losing any of those that you think. That's why you need to have the literature, and you have to study the literature, making sure that you're not missing anything, making sure that you cover everything, so that your study will be impactful, and no reviewers, no examiners is going to evaluate you such a way that ask you a lot of questions. You have to be confident. I have done this. I have showed you. I have everything. You'd ask me question, I'm going to tell you what happened to those questions, the answer, and you're passed, and you're first class. You're like maybe rank student. It's about showing that and working towards whatever you are doing. Remember, this story, go beyond. Like maybe after 15 years, 20 years, she can come back and see her work, and she'll be so happy. I have conducted all the experiment at that time. Probably in next 15 years, the technology go to the next level, and she may be having a way different technologies at that time. But still the story is going to be the same. It doesn't matter what happens, what technology comes, the IL-1 beta will be inhibited in those inflammatory markers. IL-1 alpha will be inhibited with a P38 inhibitor. Whatever she used and knocked out, whatever the data is not going to change. The technology changes so that I can use it in a different way, analyze these cells in a different way, and maybe a higher level. That's all. That's all you're doing. So our SARC EGFR, doesn't matter what, I can actually show you today with different populations showing different things. But the story is the same thing. Story is not going to change, but only that knowledge that you developed in each population, understand the microenvironment, is going to change, right? So those are the macrophage population. She also looked at the intratumoral granulocyte population. She basically looking at every population is available on those single cell RNA sequencing data. It's important because she is developing a knowledge for her future. Okay, she's not doing because mentor says, or mentor whether he says or not. She's actually developing a knowledge that's forever. So this paper is for a Bible for her, right? It's a PhD thesis. Just imagine, our PhD papers, we cannot forget. I cannot forget my papers, even doesn't matter how many papers I've published. So each figure in my mind, last 26 years, I'm still having the same memories, same figures. It will pop up every time, right? Every time it will come and go. The reason is that you spend so much of time investigating something that is for your life. You will be always thinking about it. Whenever you have a free time and looking at something, nebulin and titin, when I looked at it, I was like, I go back to my paper, 25 years ago paper, I have the PDF somewhere, just look at my electron microscopy data, or ion exchange chromatography. Did I isolate correctly? Did anything wrong? Is it the sigma, whatever the compound it was right? Is it like an ion exchange chromatography that, so the ion exchange chromatography when I did, this, I ordered the chemical, came back after 6 months. So I was like waiting for 6 months doing all this in the assay, but today I can get it right away. But what I'm saying is that those days, 6 months, probably for you guys, probably 1 month or 2 months, even now. But what I'm saying is that I waited 6 months to get that compound from Sigma from Mumbai because it was not available in India at that time. So I ordered from somewhere outside. So what I'm saying is that she actually looking at all the population, making sure that, she's getting the knowledge. She's getting the knowledge of each population. And then she was a cancer biologist. She got excited with this immunology. She got excited with immunology. She now doing the immunology. She's not doing the cancer biology because her interest was immunology. That's why she went and did all the population, making sure that if she's doing the immunology in the future, this is the starting. She wants to understand each population, making sure that she knows what they do, how they do. You know what I'm saying? You may be a PhD student, but you have to think about the future. Think about your own future, right? Your own future is in your hand. So basically in her future, if she's building right away, right there. So she become now immunologist and she's not a cancer biologist, okay? So what I'm saying is that she studied all these things, she got excited looking at all this population. Oh my God, beautiful. Like, look at this different population, how they are interacting, understanding these populations. Again, she has to spend a lot of time. I told you, it is like a tumor cell, how you study, you have to study the other cell factors too. The same way, you have to use different technology, but it's the same way you have to study it too. So she is now looking at the T cell population. In the T cells, she is looking at the genes which are regulated in T cells. For the P38 inhibition for her life, even after 10 years, she will come back and do that P38 inhibition probably, because she will see our data and see, I think this population, this gene, I think I looked at the P38, I think there's something exciting, because she saw somewhere, sometime in other immune cells that something else is happening to some of these genes, right? You'll come back and look at what happened to my own data, right? It is like that. It is that the research is always like that. Get excited and gives you confidence when you see your own data and it's happening the same way. That's why when you are conducting your experiment, conduct it right. Otherwise, you'll feel yourself bad. You'll have a nightmare later on after 10 years. Because if you're not done correctly today, and today probably you'll get away, but after 10, 15 years, it's going to haunt you. It's going to you feel somewhere guilt that, oh my God, I should have done that. I should have not done this one. You know what I mean? Things like that. So make sure that you're going to sleep well. That's why you have to do it right. So the P38 inhibition increased the total T cell population, but not the CD8 T cell activations. So that's what she compared all these different T cell activators. And actually she saw that CD4 positive, not the CD8 positive, and she was CD4 positive T cell were activation was increased, but not the 8. But that's fine. There are papers published on CD8 population increasing, some people publish on CD4 population increasing. I don't want to go in detail, but what I'm trying to tell you or give you in hint that different population have a different science, and they are even more exciting when trying to understand them, how they are actually doing it. Okay. This is another population, T cell interferon gamma production. Interferon gamma was also another key component. So people are studying at that time. We asked her, okay, why don't you look at the interferon gamma and see what happens to that production? So she just went and did all those experiments, and you can see the interferon gamma was high, which is important for the chemotherapy study. She actually asked the question whether the P38 inhibition enhances the efficacy of the chemotherapy. Again, I'm saying when she wants to take this to a clinical trial, and she has to do the chemotherapy, our standard therapy, like how we did before, we asked her, okay, why don't you try with chemotherapy, just in case if we want to take this study to the clinical trial, and she makes sure that the chemotherapy has been done right, because today's chemotherapy is a gemcitabine paclitaxel. What we have done is the gemcitabine, and then she went and did the chemotherapy study. So basically, what the P38 inhibition, and then I think the next one, I think she did the combination with the chemotherapy here, and here. This is gemcitabine and paclitaxel, and this is a vehicle. This is the gemcitabine paclitaxel plus the vehicle. So basically making sure that in the just the chemotherapy study with the knockout of a MAP kinase, which is just not the inhibitor, she did two ways. One is knocking out P38 and having the chemotherapy and making sure that we didn't have that luxury when we did the experiment. We only did with the chemotherapy plus the inhibitor, but she had the luxury, she had a knockout study. Now she has a CRISPR, and she has all this CRISPRed knockout. We have a excellent CRISPR guy in the laboratory who actually does all this gene knockout and overexpression, and she used that help and then did all this knockout. Now she can do knockout, making sure that it's not a drug effect, it's just the gene is really, or signaling is actually affected. And she also did the drug study, just not the knockout. Remember, we didn't have the luxury, she has a luxury, so she did both. She did the chemotherapy plus inhibitor. She also did the knockout plus the chemotherapy, making sure that they're all working fine. And when she did in this mouse model, so P38 inhibitor and the gemcitabine together combination working fine, and she actually went and analyzed all the IL-1 alpha, making sure that it is not something that going wrong, right? Sometimes what happens, these cytokines can be reversed after you add chemotherapy. I showed you also in our data, making sure that it is happening the same way. So that's what she did. She went and did all those. When you want to do the chemotherapy with the P38 inhibitor study or any inhibitor study, you have to make sure that tumor burden is gone, or anything that with the fibrosis, anything that in the pancreatic cancer. Maybe in other cancers differently, but in pancreatic cancer, you have to make sure that tumor burden what happened and the fibrosis what happened, right? Because of the drug delivery and all those things. And here she actually showing the survival. Our survival was much way better, but 62 days. It is not like MEC-ARC plus we did the anti-PD-1, we did long time survival. This is a 62 days survival. It is not great, right? If you ask me whether this is going to go to the clinical trial, may not. Okay, because of the survival. Because survival may not be that great, right? Maybe there's something we are missing. Okay, there's something missing. She's missing something science here. So someone will come and start looking into it. Probably we will have a survival much better. But whatever she had, she's publishing it, making sure that she is actually done well, biology is there, and therapeutically, whatever she got in these mice, she's actually presenting and showing to you. But there may be a science lacking, there may be something else is going on here. But the survival is much better than before, just the P38 inhibitor. But now she has much better inhibition, 47 days to a 62 day. Remember these mice die in 8 weeks. Okay, what she got is actually fantastic. 47 to 62 is another 2, 3, 4 weeks. That means that if you see in a human being, it is like another year or two, another couple of months probably. Okay, that's how you compare. So that is the whole story here. Basically, it's a beautiful story. Okay, how she built it, the whole story. She started here and went and did this one here, understood the whole thing in the tumor, other microenvironment. She inhibited, and she's just inhibited here also, making sure that P38 also expressed in the fibroblast cells and inhibit there, and making sure that also works or not. You know what I'm saying? So she just did the survival, making sure the survival what happens, and she did not stop. Believe me, she did another year of experiments that you are not seeing it, and she went and did a lot of other things, because it is actually the survival is better, but not that great. That actually haunting her all the time. She did a lot of other assays. I'm telling you that I'm not telling you a story, but what I'm telling you that there is a way always, because you have to figure it out. Okay, so our story, what we were telling her, our story, what happened. How I was actually explaining to you yesterday, how we went from the SARC to MAP kinase and the PD-1 blockade. I was telling you story. She knows the story, and she got excited, and she went and did a lot of other experiments that I'm not showing, right? But the research never stopped, right? She's just excited, and she did all this work. There's the AIM-1. And there are the future implications. She's actually doing all this experiment. If you say future implication, that means she has already done it. Okay, she's actually giving you a hint that I have this kind of data. She collected all this data, and she's actually telling that to you that this is a future implication, but she already collected all this information and actually working towards at that time 1 year of work, and she already knows how to improve. Okay, she already knows how to improve this from this study to the next study. Probably she's going to help next person, whoever comes. Probably she will do it herself. Okay, so there are some phase 1, phase 2 trial of this P38 inhibitor. Remember, I told you one thing yesterday. Okay, whenever you use the chemotherapy or whenever you use the therapy, whenever you use the targets, so make sure that that drug that you're using is in the clinical trial, so that it will help you to develop the clinical trial later on, right? That is the story. That's actually exactly, she's actually showing you here that drug is already in the clinical trial. She's giving you a hint, I'm going there, but she's not telling you when, right? So she just, these are all the story. I hope you enjoyed it. I think it's something that I also get excited, because when the graduate student come and start working in the laboratory, excited and passionate, right? Passionate, excited, and she's not leaving not even single paper coming out. And she's like, she says, Nag, I saw today this paper came out. Look at this one. This is what they've done. This is how they're like, I'm really excited to conduct this experiment. Important. The student and coming and telling you that I want to do this experiment, you're not telling them that you have to conduct this experiment, is what I think mentor has to train them, right? That the student will get excited themselves to come to the laboratory and conduct those experiments, and herself or himself, and doing the experiment and showing me the data. Sir, this is what I did. I was like, excited yesterday, and now I'm frustrated. Why? Because this is reason. She will give me reasons why I'm frustrated today. That is exciting data, but today I'm frustrated. Yesterday I was excited. Things like that. It can happen. But I think it's a story that I want you all listen, and so that, excited about, PhD program. If you're conducting a PhD, or even if you're doing the postdoc later on, it's fine. Or become a faculty, and if you're conducting experiment, and I think the passion, what to do, what not to do, how to do, and things like this, I think is excites more. Okay, thank you all. And if you have any questions, I'll take a few minutes. If not, I think it's a tea time, it looks like. So we can discuss maybe afternoon. We have a tutorial discussion. So thank you so much. Have a tea.
Good afternoon. I hope you had a gap in between and we can continue with the next session. So, yesterday we started with a work where we began our journey, and it is a slight continuation of the study what we did at the with another candidate. What I thought is that instead of explaining the techniques and all, let us study how the work we planned, how it was executed, and how we could finish off the work. And here you can see the title that. So yesterday we actually we worked on only on the natural products and in vitro studies and in vivo study. But here we want to combine the study with bioinformatics, and along with that, at the end we will be reaching the in vitro study. So sometimes maybe depending on your facility or depending on your strength, you have to plan the work. Sometimes it may not work in that way. So try to find out what works better for you. So in this case, so we did not work on the animal models, thanks to Dr. Nag, because this mice is from his lab. So we were studying the anti-tumor activity in the nude mice. And before going to our study, we will see why the in vitro studies, in vivo animal model, sometimes we hesitate to take that one for our research purpose. The major thing is that the time that consumes. Okay, because whenever you are going for any in vivo study, it is just like handling the humans. Okay, even they have the life and they also respond according to the way you respond to them. Okay, so they need some time for growth. They need some time for, if they are having any disease, that time you have to take care of all the things whatever you take care of a particular patient. Okay, so in that condition it is lot of patience and lot of time. You will not get the result. Today I will put all the reagents into the mouth of the mouse, and tomorrow I will not get all the results, because it should take up the drug and later it should respond to the drug, because it has its own microenvironment in its the mouse body, and that should be metabolized and so many other aspects, and it takes a lot of time. And at the same time, not only that, the labor intensive. You have to run around, you have to feed them just like human beings, they need the food, and you have to feed them, you have to give them water, and you have to make them comfortable in all the ways, otherwise they may die. Okay, so in such occasion, it is a kind of very labor intensive. You have to make sure that even the animal cages what you are using is clean, and every time you are changing that bedding material, everything, that takes you lot of time. Okay, and other aspect is that along with that, the cost of the in vivo study, how much cost it costs to carry out this study. Because if you are using the Swiss albino mice or any other mice itself, it is a very costly business. So here even one mice if you purchase, it may be in Indian rupees, it may be around 100 to 200 rupees to get a one mice. But you cannot work everything with one mice, because you are supposed to keep the control and you are supposed to keep the drug, and in between you have to carry out all the toxicity studies and drug standardization, everything. And that takes lot of cost, because even the feed you have to purchase. They have a specific feed you can, or even you can prepare in the lab itself, but it is very lengthy process to make sure that it is a properly made and it kept in a sterile condition and all. It requires, the cost will be a bit high when compared to the in vitro studies. And one more disadvantage, it is a general disadvantage. If you do even at in using animal models, you are not sure that it mimics everything in the human being, because the way mouse it has a maybe body, or its way of the interacting with the drug, or it may be its reaction to the drug may be different from how we are actually responding to the action of the drug. So naturally that makes a lot of difficulty for, that's why many drugs, even though qualify in the preclinical trials, they will fail in the clinical trial later. Okay, that is what the major difficulty is. You are investing so much amount and finally you will not end up with that. Or along with that, the other thing is that ethical issues. So you have to get the ethical permission, and along with that you have to maintain all the standards and regulations that is put forward by the ethical committee. So by crossing all these hurdles, if you carry out the experiment and later it may not completely predict what happens in the human beings. That's why we can check all those things in the animal study as maybe in vitro studies. So what are the advantages of in vitro studies compared to in vivo studies? One thing is that less expensive. You need not, maybe I am telling about not only the cell lines, there are other in vitro studies where you can cut the cost of the study when compared to the animal models. And somewhat it is easy when compared to animal models. And as I told, the animals, they need some time, because once you induce the tumor, tomorrow itself it will not grow into tumor complete size. You have to wait, you have the patience, and that will take you long time to get the results. But in vitro studies, you will get result much faster. I don't say it is completely better than the in vivo study, because in vivo study is much superior to the in vitro study. But the thing is that when you compare all other aspects, in vitro study is much easier to do. Screen large number of drugs, because when you take animal, you have to maintain certain animals, statistically it should be significant. All these parameters you have to check, and you have to check the drug on that one. So, and one more thing is that suppose if you want to check that drug further, you can reduce the number of the animals, so that the work, maybe the intensity or maybe the amount of work you are spending for in vivo studies, that can be reduced. And in another thing is that you can find out what way it acts on the cell. Okay, may not be completely, it may not mimic the microenvironment that is existing in body, but at least you can find out where exactly the drug is going and binding to the different regions in the cell, whether it is acting on the enzyme, or whether it is acting on the maybe at the DNA level, or whatever it might be, you can find out that one. And along with that, even you can find out how far extent the drug has the effectiveness. So whether the drug has any toxicity or not, that can be found out. So along with that, nowadays even we have 3D cultures, tissue engineering, and that can simulate somewhat the in vivo conditions. Okay, so when you have that kind of maybe 3D cultures or tissue engineering, spheroid culture and all, you work on that one, and if you find a very good result, then you can move on to the in vivo study. That will be much easier. And then I will come on, maybe I will switch over to the topic what I was discussing yesterday. So we ended up with this result actually yesterday. So the Mr. Anand, he worked on the isolation of the compound, and once he isolated the endophyte from the medicinal plant, he purified the compound, and then we found out that this actually acts on the HIF1 translocation. It is inhibiting the HIF1 translocation to the nucleus. So we were curious, instead of isolating all these things, because it used to take lot of time. We know how much time we spent to purify a single compound. So maybe we were actually carrying out the TLC and column, only with those two we were studying, and it used to take lengthy procedures and lot of time. And what we thought was that instead of taking the natural compound or anything, better let us go for certain other techniques where we can straight away go to the compound and then we can study. So the best that method what I found was that we can go for the bioinformatics method. Okay. So once again I coming to back to the institute where I was working at that time, that is around 2016 time, and that time Anand has completed his work, and we know that HIF1 can be the target for many of the molecules that are acting on the tumor. And that time the another person, another scholar he joined, his name is Jagadish. He actually, my way of conducting the research with the student is that first I will study what they are good at instead of what is my plan. So accordingly, I will try to design the plan so that they will be very comfortable to carry out the study in that way itself, instead of forcing something which is not working out with them. So I found that Mr. Jagdish, he was very good at handling the computer. Whenever I used to give him some work, he used to prepare a very nice slide, and the way he used to present, everyone used to get attracted. Then I thought that why, because that time also we had the power fluctuation and all other problem. Once the institute is established, maybe in as a new, it takes a lot of time to get established. And at the same time, whatever the UGC grant and BRNS grant we had had, that was actually getting over. So we did not had any new grant at that time. And the best way to come out of these kinds of situation was that we could work with something where there is no need of much investment. And at the same time, he got the UGC fellowship for doing his work. That was the one advantage. So I thought that better let him work on the aspect which he is comfortable, and at the same time I can plan accordingly so that his work will be much easier and we can reach the goal much faster. So what we did was that we thought that hypoxia would be the best factor for our study. So here briefly I will tell about what is actually it is. Hypoxic condition is a condition where there is less oxygen. So this condition actually makes us to feel uneasy. Suppose if I block all the windows here, if the oxygen concentration goes down, naturally you feel very uncomfortable, uneasiness. And we know that even if there is any building catches fire, people not die because of the fire, they die because of the carbon dioxide and carbon monoxide. So once the oxygen concentration reduces, person is prone to die. And at the same time, this hypoxia is related with many aspects. Maybe some of you might have observed also, even in anemia there will be hypoxic condition. The person will find difficulty to breathe because the less RBC cells, he cannot or she cannot get proper oxygen to the body, and all the symptoms of hypoxia can be observed. Or sometimes if you go to the high altitude also, there is a lack of oxygen, that time also you find this hypoxic conditions. Or deep down the sea, if you go, there also the oxygen concentration will be very less, you suffer from, or if you have not proper the aids, you will be having hypoxic conditions. So all these conditions, maybe that is maybe generally what it happens, but what happens during the cancer, because we are relating the hypoxic condition to the cancer. So we studied about this one, and here if you see the picture, the picture what it shows is the cells that are dividing continuously. Okay. So as the cancer cells, they are getting divided and they start getting accumulated, and as they getting accumulated, they are far away from the blood vessel which supplies the nutrients and oxygen. And as a result of this one, the cells which are far away from this blood vessel, I am not going to tell you exactly how much distance, what way, what happens and all. So if they are moving far away from the blood vessel, what happens is that these cells becomes necrotic cells. And these necrotic cells, they will not be having that kind of oxygen. And at the same time, this condition actually induces the factor called as hypoxic factor. That is called as hypoxia factor 1. Or it is alpha and different factors, they are there. And at the same time, as the blood vessels, they are far away, this cells, they actually suffer from oxygen concentrate, maybe oxygen supply, and they become hypoxic cells. And the hypoxic cells, they try to adapt, because whenever we are under stress, usually we try to adapt in one or the other way. The same way, because here the major problem with the cancer is that they are part of our cells only. They are actually kind of misguided or maybe misoriented cells. So that's why it is very difficult to treat the cancer when it comes to, except few cancers. I am not telling all the cancer cannot be treated with that one. There are certain cancers difficult because they are actually kind of maybe they are origin from our own body. And whatever we treat the cells or treat the cancer cells, same thing is taken up by even the normal cells or non-cancerous cells. And here in this case, what happens is that the oxygen gradient, it goes on decreasing as the cells are away from the blood vessels. And at the same time, if alpha factor, its concentration increases because hypoxia induces the factor called hypoxia 1 alpha factor. And at the same time, these cells, they switch over or they undergo metabolic reprogramming, what we call it as. So metabolic reprogramming in this sense, they go for more production of this lactate dehydrogenase, and lactic acid gets accumulated. Those who have studied biochemistry, you will be very much known about, maybe very much aware of this one, what happens. And as a result, the acidity or hydrogen the concentration increases or pH decreases. And at the same time, even the carbon dioxide gets accumulated and it contributes to the pH gradient or the less pH activity. And the cells, they switch over to the glycolysis because there is an accumulation of the lactate. They cannot move to the this complete, they switch over to the anaerobic and become glycolysis, and this makes the cell radio and chemotherapy resistant cells. And this makes the very difficult to treat with chemicals or even with radiation therapy, because it will not able to bring about the effect that much effectively when compared to the condition which is prevailing in the normal condition. And here this it has been shown since many years that this hypoxia is the one which actually hinders the therapy since many years. And we were studying what best we can do with this hypoxia factor, because we planned better let us work on the hypoxia factor. And then we wanted to study its structure so that we can move further, because earlier study, it was very simple. We know that the plant was anticancer, and we knew that we can isolate a compound that is which is a lead molecule, and we can go further. But here, all of a sudden, we ended up with a molecule which plays a role in the cancer. So we were stuck there, and then what we did was that just we went on a simple study. Only one thing what we did was that we were answering the question why we have to do this one. So we wanted to study, because if you have to study the hypoxia factor, you should have a knowledge about the hypoxia factor structure. This is a factor, it is actually a transcription factor. This transcription factor, once it is a transcription factor, most of the biological science students, you are familiar that it induces the expression of some other proteins. In simple terms, I can tell that one. Okay. Just like the human body, even the transcription, because I compare that whatever happens with that molecule also, they are kind of a humans. Here this, the transcription factor has different regions, like BHLH, we call it as domains. Okay. And even the past domains, don't worry too much about the domains and all those things, just see the structure of this one, because we were actually going to study all this aspect with this structure of the molecule. And then we saw that it has a oxygen dependent domain. Oxygen dependent domain is the one which determines or which senses the presence of the oxygen. Suppose if the oxygen is present, strange thing is that this hypoxia factor is degraded within 5 minutes. Suppose if the oxygen is absent, it becomes stable by interacting with other molecules. And also it had N-terminal domain, C-terminal domain. So we call this past domain as dimerization domain. So this actually favors the interaction of this hypoxia factor 1 alpha with other protein, which plays a crucial role in bringing about the cancer. And another one is that regulatory domain. Domain in the sense, those who are not from biological science background, it is a region where it is involved in certain functions. Okay, if you take any protein, let me consider if my body is a complete structure, my hand is a kind of one domain. Okay, my leg is a kind of one domain which performs independent function. That, if you remember, it is more than enough. So another one is that carboxyl terminal domain. All these domain, they are present. So what we should do next, because that's why, just to show that what are domains, I have taken different organs and they are doing independent work. So we wanted to know then whether there is a single hypoxia factor or different other factors are also involved. That time actually many this hypoxia factors, they were getting reported. So they were having different functions and they were getting reported, because we wanted to use according to our limitations, according to our strength. We did not worry too much about what is happening, because our limitations made us to think in that way. And there are different factors we noticed, that is HIF1 alpha. Along with that, there are other factors were also expressed. They are called as hypoxia factor, like 1 beta, 2 alpha, 2 beta, 3 alpha, 3 beta and all. So they had different functions and they have been reported. But we were not that much interested about the other factors, because of we were targeting HIF1 alpha. And then what we did was that because what we observed in our previous study, that this HIF1 alpha, actually it is involved in so many functions, and it is straight away it is linked to the cancer. We wanted to find out how, with how many protein actually it is interacting with. So we checked this interaction of the HIF1 alpha with all other protein. We could see that it is interacting with so many proteins that are involved in the cancer. And it is just like us, we will not interact with only one person. With simultaneously you might maybe interacting with different people, and for different topics you might be interacting with someone. Even the this hypoxia factor, it is interacting. It takes the signal from one molecule and it actually sends the signal to the other molecule. The same way what we do, they have their own network and signaling. And we wanted to find out, because this actually we wanted to study so that we can plan our work properly. We studied what happens to the hypoxia factor during the normal oxygen concentration and during hypoxic conditions. So during the normal condition, strangely, maybe within 5 minutes, this HIF1 alpha factor, it is bound by one enzyme called as proline hydroxylase enzyme, and it brings about the hydroxylation. Hydroxylation is in the sense, it is just the addition of hydroxyl group. And that hydroxyl group is recognized by some other group of protein that let us not worry the complete about what type of protein they are interacting. And ultimately what happens is that this protein is chopped off into smaller pieces. That means in the normal oxygen concentration, it is getting degraded just within 5 minutes. But when the oxygen concentration is low, it is bound by some other protein. It is just like you are having a kinds of friends where when you have enough money, you have some friends, and once the pocket is empty, you have another friend. So like that, when the oxygen concentration is high, the friends are different, even the protein factors. And here, once the oxygen concentration is low, naturally it is bound by different factors, like even the radiation causes the activation of this one. And that makes the protein to bind to certain other proteins, like the other stabilizing protein. I don't go in detail about this different factor because it may not be familiar with. There are some chemistry people are also sitting, they may find it very difficult. So here they are also binding to some other protein, and what happens is that instead of getting degraded, they are actually become stable. And once they become stable, they are bound by some more proteins. And these proteins together, they enter into the nucleus where they bind to the specific region on the DNA, where it brings about various other aspects which I have listed out here that is linked with the cancers. You can see that metastasis or angiogenesis or Warburg effect or even pH homeostasis or cell survival. So many different aspects that are brought about by one signaling molecule. That is the hypoxia factor. And next we wanted to study the sequence scientific, because there were already reported there are different the hypoxia factors are there. Best way is that if you know a person like Joshi, if you find there are 100 people here Joshi, best thing is that you find out the similarity between them or differences between them based on the height, based on the campus, or based on the different aspects. We did the same thing. We wanted to know, differentiate, or we wanted to find out the similarity between those, and we found that there are some similarities and there are some dissimilarities. I am not going in any detail about that one. And next question was that, because this is a very strange thing what we observed. We were checking, we were very curious because we were asking some few questions why. Usually people, they are getting cancer. Is there anything that doesn't get the cancer was why our major question. So anything that usually resists the cancer was the question. We started searching the literature. And as we were going through the literature, we got an article which I have quoted here. It shows that there is a mouse. It doesn't look like a mouse, but it is a mouse only. It is called naked rats because they don't have hairs on them and they actually live in the burrows, and they are actually present in a very hypoxic condition, and they live longer than the mouse which are growing in the laboratory as well as even in outside, regular mouse. And then we were very much interested because the cancer and the hypoxia may not be related in that case, because always we say the hypoxia is related with the cancer. And at the same time, there are certain organisms, they don't have cancer, but they have the hypoxia. Okay, it is a contradictory kind of the results what we were getting. We were stuck at that time. And then we studied what is the mechanism by which actually it evades that the cancer. So there are certain reasons where it produces the hyaluron, that is a kind of compound which is present in your knees where you can move your leg properly because of that lubricating kind of the polysaccharide substance. So same thing, a derivative, a large molecule that is present, that actually prevents this this naked rats not to get the cancer. Whenever the cells from the naked mole rat was grown in the laboratory, they used to produce lot of the hyaluron, that is high molecular weight hyaluron, and the cells used to be stick with each other. That's what they use a particular terminology called as goo. It used to produce a goo, and cells used to stick together to each other. And when it was happening like that, and they wanted for a study sake, what they did was that they removed all the hyaluron, and then they found that the cells were prone to the cancer. That means that even if they are in the hypoxic condition, they are not susceptible to cancer because of this particular heteropolysaccharide that is present. And not only that, we came up with another article saying that there is one more rat that is actually highly resistant to the cancer, and it lives longer compared to the other normal mouse. So that is called as blind mole rat. Okay, and here the mechanism is totally different when it comes to the blind mole rat. It is also highly resistant to the cancer, but because of the presence of DNA methyl transferase 1, it can prevent the cancer spreading. That's what the article says. And these two are the organisms which we got the information that they are highly resistant to cancer and they live longer. One is the naked mole rat, and another one is actually the blind mole rat. And then we had a question, because if you look at the conditions, the oxygen concentration is less even in depth of these oceans and at the high altitude also. So we will carry out a study where we will study the relation or maybe the correlation of this hypoxia factor in different organisms. They are they are living in different atmosphere, whether it throws any light on our study. So we wanted to select the organism. Our idea was very simple. We selected few organisms which are related to the human being, because we were looking for the bioinformatics study. So we selected the organisms like human, chimpanzees, maybe this orangutan and monkey and all. And along with that, even we selected dog. And at the same time, the other animals we selected, whether it is cow or yak, which are in the maybe Tibetan antelope, which is actually living in the high altitude, to get an information. And at the same time, we selected even the lab mouse also, okay, regular normal mouse, and even the rat, and we included these two, because earlier study we did not included these two, because we did not know that there are certain organisms, they can survive even at very low concentration of the oxygen. So we selected that one, and then we selected the two organisms. They are surviving in the water. They are actually having the hypoxic condition there. So our intention was very much clear. We have taken almost all the organisms. They are representing the different atmosphere where the hypoxia factor or hypoxia factor is getting expressed. So once we studied that one, we searched the sequence of the hypoxia factor of these 16 organisms. And the best way to compare them was that, because we did not had this lab to check the in vitro or in vivo type of studies, what we did was that better let us study with the computer itself, whether they have any relations or not. So we found that best way is that you can carry out the multiple sequence alignment. Most of the research scholars, you know what is multiple sequence alignment. You just take the sequence of the DNA, you match with the other DNA, you find out how much extent they are similar or dissimilar. This is the basic principle. And we found that there are certain differences, there are certain similarities. I am not going in detail, the technical aspect. We found that there are certain variations in certain region, and there are certain maybe similarities in some organisms. And these results, even we took, there are certain domains also, and we saw if there is any similarity and dissimilarity, and we found that we could arrange the organisms according to the hypoxia factor, the relatedness with the hypoxia factor. If you look at the human beings are related with the chimpanzees more, and they are related even with the orangutan, maybe with related to hypoxia factor I am telling, okay, nothing else about the evolutionary history, just I am telling with respect to hypoxia factor. And even we could see that all these yak, cow and Tibetan antelope, goat and all, they belong to the same category. And this blind mole rat, and interestingly you can see that the blind mole rat and mouse, they are at the one category, and this deviated, this naked mole rat, it deviated with its relation or similarity when compared to other. And as expected, this naked carp and zebrafish, they were in the separate clade. What does it indicate? So this indicates that it is a kind of evolutionary relationship between the hypoxia factor, just like the humans evolved, along the way even the hypoxia factor evolved. And then we wanted to study, because I was telling about a domain or a hand which is dependent, or its stability or instability is dependent on the oxygen concentration. We wanted to find out how far extent this oxygen dependent domain is related with each other organism. You can once again, you can see that they have certain relations, and there are certain organisms, they moved out of the clade, and there is a relationship between even the oxygen dependent domain also. And then was that we wanted to find out, because the blind mole rat, which is a highly the cancer resistant, or even in hypoxic condition which cannot get the cancer, and the humans, we are naturally prone to the cancer. And this HIF1 alpha domain of these two organisms, do they have any relations or not? We found that there are certain key residues which I have marked here, they were one and the same. And there are certain residues where there was a difference between the humans and maybe this blind mole rats, indicating that maybe these residues may be playing a role in the way organism it responds to the oxygen. So because the candidate was enthusiastic, and this study seems to be very simple, but it took nearly around 6 months to do all these things, because he was supposed to learn all the bioinformatics tools. And then we came up with a paper saying that what is actually the sequence analysis and phylogenetic studies of hypoxia inducible factor 1 alpha. And then what we did was that now we know that we are very much sure. So there is a relationship, and there is a dissimilarity between the organism with respect to the hypoxia factor. Then what way approach this hypoxia factor? Can this hypoxia factor be the target of anti-cancer drugs? Because we did not know which drug will actually bind to the hypoxia factor. So for that, we started using the bioinformatics. Instead of taking one molecule, and at this end, if you end up with none, it is waste of time for even the research scholar, and it is waste of energy. So what we did was that instead of doing like that, let us create a maybe in virtual condition where the hypoxia factor will be selected, and a drug will be selected in such a way that it will go and bind to that one, and it prevents its interaction with other molecule. Because earlier itself I was telling, the hypoxia factor is binding to the different proteins, and those proteins are the one, they will determine whether this hypoxia factor to be degraded or it should be disabled. So that's why we wanted to study, is there any molecule which can prevent this protein-protein interaction, so that we can target that one and prevent the stabilization of hypoxia factor, and in turn we can treat the cancer. That's what our hypothesis was. And for that, what we were supposed to do, I am simply telling about what actually we did. I am not telling each and every technique what we are doing. And we took the receptors, because receptors also we are getting maybe we have to build the models, because unless if you don't have the model, you cannot bring the drug and you cannot target it to the models. So we were supposed to build the model for that hypoxia factor. So we took the PDB, protein data bank, and we modeled using various softwares. And not only that one, there are different ways we have to authenticate that one. So either it is pre-processing or grid generation and all, let us not worry too much about that one. So ultimately what we did was that in our study, we prepared a model which can be docked. Docked in the sense, I can bring a drug and I can put it onto it, and I will study whether it interacts with the other proteins or not. So we did that one, and even the ligands, if we have to select, they should be selected in such a way that there are certain rules we are supposed to follow. They should have certain amount of size, that is, we call it as Lipinski rule of 5, and if they actually qualify that one, then we will go ahead with that energy minimization, and all other studies we will do. It is just like the way, if you start writing, you need the pen, you need the paper, you need something else, you need the chair, and if everything is proper, then your study goes fine. So if the chair is not proper, you will make sure that that chair should be proper. In the same way, we made sure that different techniques we have used, and we made sure that whatever you are doing is foolproof. And then we analyzed, and later on we actually visualized the molecule, how it acts. And we selected different domains, that's what I was telling, various domains, and we have used different the software packages to consider this PASS domain or BHLH domain or CTAD domain. Don't worry too much about this one. And then even we studied whether it is the dope or dope score model or even Ramachandran model to establish that it has a proper way of arrangements. And finally, we wanted to target HIF-1 alpha factor. And in HIF-1 alpha factor, as I was telling, it has different domains. So whether I should target that domain or this domain, we had a doubt. So we went to the literature. When we were studying, we found that the PASS-B domain of HIF-1 alpha is majorly involved in the interaction with the HIF-1 beta. And to become along with that, the PASS-B domain, it also interacts with another protein that is called HSP90. Most of the biology students, you are familiar with this HSP protein or heat shock proteins. They are actually considered to be the molecular chaperones. They maintain the structure of the protein. Okay. These two proteins, they were actually interacting with the HIF-1 alpha protein. So here, the HIF-1 alpha beta interaction is necessary for the entry of the hypoxia protein into the nucleus and bring about the activation of many genes which are responsible for cancer. And the HSP90, it actually stabilizes in such a way that the HIF-1 beta can bind. So these two factors, actually the key factors, they are making this HIF-1 alpha a strong molecule so that it induces the cancer. So then what we did was that we analyzed what are the binding site, because if you shake hand, you know what are the regions actually interacting with one another. Just if you put some block between the hands, then naturally there won't be any interaction. That's what our the plan was. So HIF1 alpha, HIF1 beta interactions, they are mediated through some key amino acids. So if you can block the interaction between those key amino acids, naturally there won't be HIF1 alpha, HIF1 beta interactions, and naturally that the HIF1 alpha is taken away by the degrading enzymes, and it is completely degraded. So then we wanted to dock with compounds. That's what I was telling. Instead of taking one molecule, why can't we select too many molecules or library of compounds and we screen that one? Because we did not had the facility, we can't sit quiet. So we had the computer and we could connect it to net and we could do some studies. We thought that better we will go ahead with the library screening. So we selected nearly 67,823 compounds, and then we applied the Lipinski rule of 5. I am not going in detail about all these rules and regulations. They actually, one thing you remember, this rule implies that if the 5 rules are followed by a compound, that compound is considered to be the drug. It should have a molecular weight between 500 Dalton, something like that, or it should it should have a capacity to cross certain region, all those things. And this led us to the key down to around 2,753 molecules. And later on we actually further screen the compound, make sure that whatever we are doing the correct study, and we created a compound library, and this compound, from the compound library, a pool of these compounds we have selected, and we wanted to dock and we wanted to find out, and then we carried out the docking study with various softwares what I have mentioned here. And this thing is that actually Schrodinger, we had an advantage. At that time only this Schrodinger software was purchased in the Department of Industrial Chemistry, and we actually took the help of the industrial chemistry to carry out this Schrodinger, maybe to carry out this docking study and other studies using Schrodinger, and we could get a better result. And when we analyzed this result of the compound library what we prepared, we found that there are certain compounds. Usually whenever in the bioinformatics we have a practice, we will study what is the binding energy. The best way to find out whether one molecule will bind to the protein in a proper way, whether it interacts with the proper way, best thing is that you go and study what is the binding energy. So if the binding energy is maybe it is shown in the minus, if it is the minus value, if it is high, then it indicates that it has a very high affinity to its target. So we studied nearly, here I have mentioned about 13 compounds, and these 2 compounds we found they are attractive when compared to other compounds, because we cannot carry out all the studies using all the parameters, because there are different hiccups between them, because each work it takes a lot of time to do that one. So then we docked it and we studied whether the molecule really it is going and binding to the drug. And we found that of course they are interacting with the drug, and they are interacting at different regions of that particular protein. So I have mentioned here different amino acids. They are the key amino acids. They are actually interacting with this particular compound. We called it as D1. This is the compound. And later on, the same thing that we studied with another compound, that is actually the 2 compounds we selected, and this one was also actually binding to PAS-B domain. So PAS-B domain, as I told earlier, is the domain which is necessary to interact with the HSP protein as well as HIF1 beta. So if I can show that it is binding to the PAS-B domain, it is indicating that it is preventing the binding of the other protein. It is just like if you are holding one hand with maybe your friend, at the same time you cannot hold the other person's hand. So same thing we assumed, and we thought that these two are the better one for further studies. And later on, we did not stop there, because one is that we wanted to find out what way this HIF1 alpha and HIF1 beta interaction can be blocked. On the other hand, we wanted to find out how HIF1 alpha interacts with the HSP90, because HSP90 is the one that actually it mediates the stabilization of the HIF1 alpha factor. So we wanted to find out what exactly that interaction is altered by our molecule, and we did the similar studies to find out what way it actually prevents that the HIF1 alpha HSP interaction. We found that there are different molecules, they are also preventing this HSP as well as HIF1 alpha interactions, that is the protein-protein interactions. And these are the studies that are showing how exactly these molecules, they are interacting with the protein. So there are different compounds, I am not going in detail about that one. And when we were studying this one, the advantage for me was that if I tell one word to him, he used to find out 10 different possibilities of that work. And he was actually studying, we were actually focusing only on HIF1 alpha, HIF1 beta and HSP90. He wanted to find out what is the effect of this particular protein on different other protein. That time there was a new protein that was developed or maybe reported that was called as Tigar protein, that is TP53 receptor protein, and that protein was actually involved in the oxygen gradient and all, and that is somehow linked with the cancer. He got a paper and he was very curious to know what way this hypoxia factor that actually regulates that protein also. So he started studying about the, once again we constructed the model and we wanted to find out what molecule that binds to the molecule, and later we wanted to find out, because this Tigar protein, it actually interacts with phosphofructokinase B2, and we wanted to find out how they interact. I will not go in detail about all these aspects. And later we did the simulation study. In bioinformatics, one thing is that whatever you predict may not be right. You have to mimic the condition that exists in a natural way. That we call it as simulation studies. So once we did the simulation study, we found that this protein also interacting with the HIF1 alpha. With that one, we could propose our hypothesis saying that how this Tigar protein with the maybe hypoxia factor, they can be the key regulator in apoptosis regulation, and it got accepted. So that means our hypothesis, we assumed that it is right. And then once we knew that there are certain compounds, they prevent the protein-protein interaction, and we got the information from our study that they can be the best drug. And that we wanted to cross-verify. The easiest way was that the cytotoxicity assay, because that's what we did. So here we usually, those who are not familiar, because there are some MSc students, yesterday also I mentioned what is cytotoxic study. We will take the cells and we will detach these cells which are grown in the culture, and we will take in a plate which contains, we call it as microplate, and we add the drug and we incubate, and later on we find out the absorbance and we will find out what is the action of the drug. This is the simple theory behind cytotoxic assay. And when we did that one, we were thinking that if it is supported by our whatever we did in bioinformatics by the cytotoxic study, then we are right. So we checked with different cells, and then we had a question: which cancer cells to be selected for these studies? Because I cannot randomly select the cancer cells and I cannot say this is acting on HIF1 alpha. So because I wanted to show that the hypoxia factor is increased and it prevents that one, and even at the same time it prevents the cancer. So we searched the literature, we found that there are certain cells where hypoxia factor is produced a lot. Maybe one of that one is that breast cancer, as well as even in the prostate cancer, we found that the hypoxia factor is expressed in very high concentration when the tumor is present in a patient. So we thought that let us select the same cancer cells and we will find out the cytotoxicity. Of course, we got the cytotoxic activity of the drug, and when we found, when we compared this one with the non-cancerous cell, it was not showing that much activity. That means it is not that much toxic to the non-cancerous cell when compared to the cancerous cell. The same thing happened with the other drug what we did. That drug also showed a similar trend where it was active against the cancerous cell, but it was not active against, maybe lesser active against the non-cancerous cell. And I told you, because we wanted to show that experimentally that it is actually acting. Just we wanted to show that the molecular mechanism also, that it is preventing the HIF1 alpha, HIF1 beta interaction. So we wanted to find out how exactly it is actually preventing protein, maybe alpha beta interaction. We clearly showed that whenever, and here one more thing, we had a problem with the generating hypoxia for the cancer cells. But we searched the literature whether we can do any other method, because hypoxia chamber is costly. We could not afford that much. So we did not have the fund. We wanted to find out, is there any alternative way we can artificially create an atmosphere where it mimics the hypoxia, which creates the hypoxic condition. And we found that this cobalt chloride could induce the same effect, and we used that one, and we studied what happens to the HIF1 alpha expression if we treat the cells. And we found that whenever we treated the cells with the D1 or D2 drug, we found that actually the expression of the HIF1 alpha, it is coming down, indicating that whatever we hypothesized, that is actually working out. And then even we did the immunofluorescence study. It looks blank, but in my computer it is showing, it is not a good picture. And here also the immunofluorescence study, we showed that whenever there is the drug D1 and D2, it is actually preventing that the interaction of the HIF1 alpha and HIF1 beta. And based on our study, we designed a model. What is actually the mechanism by which the drug is acting? And here we found that the drug, whatever we had selected, this prevents the HIF1 alpha, HIF1 beta interaction. These are the two drugs. And there is another drug which acts on, we studied even the tiger, and it acts on this particular region. And the other four drugs, they are acting the interaction between the HIF1 alpha as well as HSP90. So all these suggested that suppose if we block that HIF1, maybe the protein-protein interactions, that protein-protein interaction definitely has a good role in bringing or reducing the cancer. That's what our hypothesis. And then we studied further, because if we want to continue that study, we studied with various other aspects. Just I will, for your sake, what are the limitations of
in very detail, or superficial about the therapeutic strategy in cancer research, how to design. You probably, have seen from last 2 days the many technologies that we have presented, and the in vitro assays, not so much in vitro, but in the today's presentation morning, I think you've seen a lot of in vitro studies. I think important is that many in vitro technology, I can say many technology, but might not work in each and every experiment, or the ideas that you're carrying to conduct your research, right? Sometimes you use this in vitro assay, sometimes you may not use that assay, sometimes you use something else, okay? So they were asking yesterday that what do we do with the drug when you start the work, right? You do the proliferation, invasion, metastasis, not metastasis, migration, colony formation assay. You can take the 3D cells and also culture, 2D cells you can culture, and do a lot of things. These are kind of a smaller, invasive, all these things. It's important experiments, MTT assay, viability assay. There are many assays you can do. These are all very important. And also in each one, you get some information that is useful to a lot of these things, right? So, but those are important, not enough, right? Not enough means that if you want to develop something, if you want to do something greater, probably those assays not enough. Probably you can publish a small paper and showing that this drug does this, and this is a natural compound, this is something you're working in your laboratory, or or you want to do something in a PhD program. It's enough. But if you want to do more, you have to bring the technology. You have to design your experiment differently, right? For example, I showed in the morning many of those in vitro assays she has conducted, right? Probably she might have conducted even more. I only showed you some of them. She might have put all the data in supplementary. If you go see the paper, probably you see more in vitro assays that she has done supplementary. But even though she has done many of the in vitro assays just in the main paper too. So, while designing therapeutic strategies, you have to know the technology. If you don't know, if you have not conducted, you should know your colleagues or someone else in the laboratory or someone else in the other lab, somewhere in the other university, and carrying or learning from them also important, right? Talking to them and also knowing what other experiment are, how they are conducting, what they are conducting, even the other laboratory. For example, you are from chemistry, you have to talk to a botanist or the zoologist or immunologist or some other biotechnologist, because some of the technique that they use, and probably it may be very helpful to you, right? That's, I think, key important. So what we're going to do now in this session, we try to understand the rationale behind the cancer design, like how you design your therapeutic cancer therapeutic strategies, and explore some of these methodologies. Already you know all the methodologies. I presented many things. You know the already concept, the tumor intrinsic, tumor extrinsic, all the work and the tumor microenvironment, kind of you touched everything. But here in this one, we just do the same thing in another way, another parallelly, something very different. Also, we'll discuss the translational clinical challenges that we're going to face, or the trials that we ran. So the example that I have given trials, for example, clinical trials, is for the phase 1 trial, right? There is a phase 2, there is a phase 3, and FDA approval of the drug. It's a big $1 million question, because they spend a lot of money. But from the phase 1 to phase 2, phase 3 is a lot of money, right? When from phase 2 to phase 3 is again a lot of money. But that investment, I and you will not be able to do it, okay? Someone else from the company or from the university maybe make a company and do it, or someone else will do it. But probably we will end up in phase 1 trial most of the time, right? But if you join a company, but you will see how the phase 2, phase 3 trials will be conducted. Probably they will be conducting, and you will know what are the parameters that you have to do and things like that. We're going to touch a little bit of those, And also important is molecular basis of cancer. You have to know the preclinical models. You have to know the molecular basis of a cancer, which cancer you are studying, and also the molecular infrastructure is different in each cancer. You all know already. So I have touched the pancreatic cancer, and the same way I have also explained the other cancer. What are the differences, like mutations or any other regulator? For example, I was telling the other girl that when you take the calves or the fibroblasts. So in pancreatic cancer, fibroblast cells look the same like other cancer fibroblasts too, but those are the structural proteins. But if you go inside, the signaling probably will be different. The receptors on the top probably different, and the target may be different. And inside the genes regulate. For example, the Samara isolated all these calves, and I showed you. She isolated individual, all this population of the calves, and then she did all this sequencing of those. What are the genes are there inside and all. Those genes, for example, if you take out these calves or the fibroblasts from other cancer, like for example lung cancer, probably those signaling pathways may be different than this one, but maybe structural proteins are same. That's why the structure of the fibroblasts will be same. So these kind of things, the molecular basis of each cancer is different. In each cancer, the population of any of this microenvironment, any of this cell population is different. So you need to understand that before you carry preclinical models. When it comes to preclinical model, see, for example, I use the KRAS mutation, P53 mutation. In lung, they may use it differently because the mutation in the RAS is only the 40 to 50% in lung, and there are other mutations maybe. So you use the model according to the mutation. And what is that important? Initiation of the cancer. In each cancer, the mutation profile is different. So, but the technology is same. The way that we conducted in the pancreas is the same way you conduct in other cancers too. So what I'm saying is that preclinical models that you use to study in different cancer is different. They have different models, but study the same way. Okay, you have to isolate all these populations, study the tumor intrinsic. We have to see the molecular level, cellular signaling, and all those. So when it comes to combination therapy, combination strategies that we did in the pancreatic cancer, you saw all those how I did yesterday and today, and the same way you design in other cancers too. But in some cancer, probably targeting a single drug may be okay. Okay, because they're all not tough tumors. Okay, and some cancers you may have to do the combination therapy, and with the chemotherapy, with the immunotherapy, like that. So in melanoma, you have to do the immunotherapy because it's very important to do the immunotherapy like that. So you have to know, like, what are those, bases, that strategies that when you take different cancer. Precision medicine. So this is like huge thing, right? Nowadays, everybody is talking about precision medicine. They are talking, but important is that whether how you conduct precision medicine, or you have to collaborate with someone, companies, or someone who is actually doing those kind of methodologies in their laboratory. So we touched a little bit of this molecular basis of the cancer. Overview of oncogenic pathway means that in some cancer EGFR mutation is important. In some cancer P53, some cancer KRAS. So when they analyze this in this paper, the Cell paper, the 10 cancer pathways. There are 10 different cancer pathways. In each cancer, different pathway, but they're all pathways though. Okay, and also there is when they analyze all these things, they got into like brain has like 628 some in that different mutation, different copy number, methylation expression. Though in each cancer, if you take a leg, something cancer here, so they have a different signaling and different things. Okay, that's I think important to you to know that they are different. And also when they did the drug study, for example, 100 plus cancer drug studies, and where they saw how this combination work, how the synergy of this combination work. Okay, all single drug, how that is going to work. So this information is very important for you. Okay, there is tons of data out there. This is 2018. Remember, 2018 to today is almost 7 years. Just imagine over this one, what and all the people published. Okay, amazingly, there are tons of data out there that people publish by using this type of technology. They bring another technology ideas. So you have to have those literature with you, understanding it. If you cannot understand, try best to talk to someone or communicate to someone and try to dig more and more. Don't leave it, because maybe you're losing something very important thing in that paper, right? It's very important. And also we talked about genetic and epigenetic alterations, and these are all the same thing, which we discussed during last 2 days. Driving the tumorigenesis means that, again I'm coming back to RAS and 53NE mutation. Tumor heterogeneity is and its implications. So now you saw our own study yesterday and today, when we talked about tumor intrinsic targeting these drugs are not enough. Then we went on and did the immunotherapy. So means that you have to understand the tumor heterogeneity. If you don't understand those heterogeneity, like we were not able to, because there was no technology to understand. Now today you can understand it. You can isolate those cells and do a lot of different things, and or someone might have already published, understand from the data and see what this population is doing and where the genes and which one is important. Okay, if we can understand, in our own study we were able to understand, and then our survival was boom, like unimaginable survival we got. So the same way you're missing something. That's why you need to understand the tumor heterogeneity. That's why people are saying, please go understand heterogeneity. Then you can implicate your cancer or the therapy better, right? So validating the therapeutic targets, right? So how do you validate the therapeutic targets? For example, somebody asked me yesterday that a different concentration of the drug, or how do you actually think how much to use in vivo, how much to use in vitro, these kind of things. One thing that we used to do, we used to run, because I had a MD chief that also worked with MDs, so they knew clinically how much they are using the drug. For example, dasatinib, that's a SRC inhibitor. My mentor, we knew how much they are using in this clinically, in a SRC inhibitor, because it was clinically approved. Remember, I always said, you use the clinically approved drugs if you can. If there is a new target you found out, probably there is no inhibitor, it's okay, but you are actually finding it. Probably you end up publishing it, not going forward, because that is not clinically approved. If you have to get the approval or doing something, you have to be attached with a clinician or medical oncologist and conduct those kind of experiments. There are a lot of companies in India actually just do their clinical trials, right? There are many companies. You can actually talk to them and see whether what they are doing, what drug they are using clinically, and you can actually attach to them, combine. They will love to have the collaboration from academia. Only thing is you have to go and attach to them, because you want to understand what they are using, and they don't want to be with you because you're still doing the basic science like 10, 15 years ago. They are beyond, right? So you have to be in their level and to compare your study to them. Then they will come to you and they want to use your data and they want to combine and collaborate with you. So your science has to go to next level. Then only they can come, right? You have to develop those kind of, for example, you have to know what is orthotopic model, what is xenograft, what is PDXs, what are those organoids, what are the spheroids, how do you isolate organoids, how do you make the spheroids. If you don't have those kind of knowledge, if someone is talking from the company, probably you will not be able to collaborate with them. So you have to develop those knowledge, and also once you develop the knowledge, go see if someone is conducting this kind of experiments. Nobody says no to you, right? So there may be other university, there may be other, like you all came to this course, you're learning something. For example, if you would have not, probably you would have lost something, like not able to, right? You're maybe a cancer biologist and you're maybe you're isolated in somewhere and doing, probably not getting exposure of something, these technologies and methodologies. Probably this is one platform that probably you're getting ideas from someone who are really doing their cancer biology from last 25 years. It's important. If someone is giving you a hint, yes, even though it is a small thing, doesn't matter where, you have to take a bus and go go meet them and see what they are telling. Probably there is always something gives you an idea. For example, in Vanderbilt, I think there was a Nobel laureate. I always, when they have a Nobel lectures, I just go, because it is not for what experiment they have conducted, it is the experience what they went through to conduct those experiments, right? For example, there's someone is coming a Nobel laureate somewhere, right? It's not that you're going to see the Nobel laureate, it's about the experience of what they went through to conduct those kind of experiments, and that actually inspires you to do something better in your life, right? It's not that Nobel laureate, like there may be someone who is pioneer in one work, pioneer in some research, a chemist or someone developed the drug who are actually giving you a talk. Probably there are some inspirational talks. Plus, more than that, there are some technology-oriented investigators. They give you a technological talks, which actually might help you. But you might be not doing the technology, but you're getting the idea of those technologies itself is actually a huge thing for your research, right? You may be like, working in the laboratory for so long, but you will not be able to, understand what's going on. But sometime when you go out like this, probably you get an idea. I think that idea might change a lot of things. That happened to us. That happened to us many, many, many times. So I think it's a key. For example, someone is presenting from the pediatric cancer. I have attended pediatric cancer, but my lab people says there's a pediatric cancer, I don't want to go. I know what they are thinking, but my thinking is different. But if I go there and see, maybe probably one day I will understand what is pediatric cancer is. So what is that they are actually going through? What these actually kids are having a issue? Why these kids are getting cancer early on? So that itself, someone is presenting who is experienced and who knows exactly what's going on, and that experience that actually you're getting that information for yourself, understanding that biology probably help you how to do it in your. For example, the cancer, pancreatic cancer. So it's pancreatic cancer kind of similar to a prostate cancer, right? Probably you might not know, but I just understood from one another talk, who is expert in pancreatic cancer is called David Davidson. So he was actually talking about pancreatic cancer biology probably related to prostate cancer. I started digging all this prostate cancer biology. I went to these talks in prostate cancer biology because that is actually giving me a hint how to study my biology, my cancer, right? Some hint because each cancer is different, but some cancers are all related. Means the way that they develop into cancer, right? Some of these high throughput screening omics technologies are important. Okay, when it also depends on kind of drug study or why you want to do the high throughput screening and what is your intention. Intention is to investigate something new, and yes, you have to do the high throughput screening. Your question was something related to developing some small science. You don't need a high throughput screening. All you have to do is dig and something literature survey and collect all this information and do it, right? It's all about what you want to do, right? And using the bioinformatics and genomic databases, for example, TCGA have written, but there are a lot of bioinformatics tools available nowadays, right? There was a student I talked to, she's from Bangalore, and she was doing MS program in Boston University. When I was talking to her, I was amazed to know she's from Bangalore, but what amazed to know, so I just got in contact. She was actually searching for internship, and I asked her, what are you doing? She's exactly doing what I wanted to, someone bioinformatician to do. I was like amazed to see, sometimes you never know, you cross with someone, and that person is actually doing something that exactly what you may be doing, right? It's just happened to us, and I want to hire her. It is like that. So we're going to hire her. So because that is exactly what we wanted, particularly right now what we are going through, right? Similarly, sometime what happens, what you cross, and when you're talking to another person, nowadays phone is an issue, right? So we will be on the phone, we will not be able to talk to anyone. Sometimes I think better to talk to something, better to leave the phone and talking to something. Probably you get a lot of information. Sometimes you have many questions will be solved, right? It's just a human nature. The technology you use for validating a lot of assays. For example, you use cell viability, apoptosis, proliferation. These are all in vitro, in vivo xenograft, genetically engineered mouse models. Like that, many models are available. I don't have to list out all of them, but important, I think, the concept and ideas that you develop to use those models, how to use. For example, you may not be able to use it here, right? Someone is already doing it. You can collaborate with them. But your science has to go to that level, probably to collaborate also. But your science the in vitro assays and in vitro studies has to go to that level. Then only that person will come and tell you, because that person is actually working on the in vivo and he will not be able to work on the in vitro because there is no knowledge. There is no person, there's a labor work, like a lot of work in vitro. Actually, more than in vivo, I think you work in vitro a lot because there are a lot of variability. Today you go to cell culture and you conduct the cells and all those things, and then you go after a week, probably your data will be different. Even the same cells, same culture room, it can happen. There are a lot of variability are there in vitro cells, and then you conduct one of the qPCR, and then after some time, probably you may not get the same fold change sometime, right? The variabilities are there. It's very common, but important, I think, validating those parameters, right? When it comes to a preclinical model system, right? During the drug development, you use the cell culture, which you all doing probably, and there is a 3D organoids, because if you want to study the microenvironment, if you want to study how this drug is transferring through the membranes and how they, cell attachment on all these microenvironment, you have to create a microenvironment to the cells. You have to use the 3D organoids. 2D is not going to be enough for you, because your 2D, you're not going to get all those information. It all depends on, again, your scientific question in your study, okay? That study question should take you there, okay? And in vivo models, all these models we talked about. So when you're using in vivo model, xenograft, so why you use xenograft? Probably if you want to just to know what happens to the tumor weight, tumor volume, that's all you get the information. You cannot take the xenograft tissue and study all this immune other population. No, you're not going to get the immune population. They are under the skin. You're not going to get all those population because the tumors are not in the right spot. You have to have those tumors in the pancreas or the lung or the lung or in brain or wherever that's supposed to be. Then only you can study the tumor microenvironment, right? You're studying the wrong model. Many people published in a xenograft and then take the immune cells, and I've seen many papers. But if that comes to a review in my table, probably I will reject those papers, right? So it's exactly because that is not a right model. So like that, you need to know what these technologies do, but that you have to develop a knowledge. That knowledge is just from the publication paper is enough. You don't have to talk to someone. It is available already. You need to know how to search and get those information. Searching is a key factor. Searching itself is 50% of your research is done. All you have to do is another 50%. If you don't know search 50, that searching, right, in the literature searching and right way, okay, it will be very difficult to do research, good research, right? You need to develop that searching, and you learn from yourself though. There's not someone is not going to teach you how to search all these things and get. You fail and you search again and you learn something and again you search, right? There are some basics you can get it, but what I'm saying is at the end of the day, you develop your searching knowledge yourself, that how to do better, okay? And there are challenges, there is no doubt. The reproducibility issue in science, right? In your own biology or technology, there is a reproducibility issue. Everyone is facing, right? So it is very common. But the way I have explained to you last 2 days, like we don't have the reproducibility issue. That is the reason that they went to a clinical trial. There is no way we have an issue in the reproducibility. The reason I will tell you, it is the way that you model your study, the way we validate every step. There's just not one or two, there are 3 or 4 times you have to modulate your study, right? For example, if it is not a KPC mice and the PKT mice, you take another mice and probably study, probably there will be a problem or maybe a issue. But the model that I have showed you and studied, those models will not have a reproducibility issue because that has been conducted in many times, conducted from different ways, making sure that is actually working. So it's, I think, important as a scientist that you have to develop the study, a reproducible study. I think that's a key factor, right? There are these things so that each one, whatever I'm just saying, this is a PDX model here, is like testing, how to test, and all those things. But it's actually a nice paper here. It's actually very recent, 2023. I loved this paper, so you just actually see what you can do. When it comes to drug discovery and screening approaches, right? There are a lot of people high throughput screening they've done. So for a small molecule library, which I'm going to explain to you tomorrow, when we do the natural compound screening and stuff. And there is a new technology, RNAi screening, CRISPR screen. These are all new things. Believe me, there's like fantastic data. I think it's all about what you want to do, how what's your research, okay? What's your plan? It's all about that. So I did not show any of those screenings so far, but I'm going to show you a little bit tomorrow. But the important, I think, why you want to screen, why I screen those compounds, I'll tell you that story tomorrow. But I think your science has to take you. So your problem or the questions that you have in your biology should take you whether you should screen or no, and what for, and what questions. So we'll discuss that tomorrow. So another optimizing the preclinical mice model or the preclinical studies. I showed you today morning that how did we optimize? We optimized using different model system, but they are all the same. They progress and become a cancer. And also we optimized with the orthotopic model and the xenograft model and PDX model. We want to compare with the human tissue and compare that data. And also there are a lot of organoid models that I did not show, but there are a lot of organoid models we have developed, spheroid models we have developed. So you have to show that indifferently optimize all those studies and making sure that preclinically you are stronger. The one another thing is when you are doing the study like all this chemotherapy, what I did not show you is that pharmacokinetics. How did we do the pharmacokinetics? And why we did the pharmacokinetics? How did we analyze the toxicity and the measurement of the toxicity? And what parameters did we use to measure the toxicity? Those kind of things. These are all done by our a team of pharmacologists and a team of pharmacokinetics people, a different group. Because remember, your expertise are different. Our expertise are not pharmacokinetics analysis or toxicity studies. Only we understand and understand those biology and things. But they are the experts. They just run those experiments and regularly. I think there are a lot of, in India, there are a lot of pharmacokinetic studies conducted outside small companies, outskirts in Bangalore. I've seen many. So it's like that. You can actually, you have to get some money and go there and conduct those pharmacokinetics because they're experts. They do. Once you know how they have done, probably you can manage to do yourself or with someone, someone colleague or other university, someone collaborator. So I think it's important to use the expertise when you are doing the preclinical development, especially pharmacokinetic analysis, toxicity measurement, right? When it comes to a combination therapy, when you want to add drugs together, right, and overcoming therapeutic resistance. Say for example, I showed you example SRC EGFR inhibitor. How did I do it? On MEK STAT3 inhibitor, how did I do it with adding the immunotherapy or immune checkpoint? These are some of the examples just I showed you. But that was conducted 10 years ago. Those experiments were conducted 10 years ago or 7, 8 years ago. But remember, today what we are doing is entirely different. The combination therapy and merging together two drugs, we have a lot of technology today. Okay, we didn't have that at that time. So the same way like how I'm talking about single cell sequencing, single population, different population, the same way when it comes to a drug study, there is a new technology, synergy study. And you don't have to worry so much like before. We don't have to go wrong. There is technology developed. So you can actually use those synergy development and also overcoming resistance and all those things. So I'm going to, I think, hit that in the next 2 slides, and I can show you a little bit maybe there. So, and also when we are doing the preclinical development and the post-clinical, first after phase 1, right? You have to do the biomarker analysis, making sure that the study, whatever you are doing, are conducted in the preclinical or the post-clinical. I was talking about the pharmacokinetics analysis and the toxicity analysis. You have to do those, monitor those, analysis, and I'm not going in detail of the what analysis you have to do. I think you can actually get it, in literature survey. And important, I think you have to do those toxicity analysis is either in the preclinical model or in the post-clinical or after the phase 1, after the patient data that you collect. Remember, if you are collaborating with a medical oncologist and if they are ready to take your drug to study in their clinic, okay, believe me, you get all those data because your study, they are actually doing it because they believed in your work. But they will give you the data. They will give you the blood samples, tissue sample, everything. You can do a lot of toxicity studies, a lot of different biomarker study that actually helps you to develop a phase 2 trial. Okay. So when it comes to preclinical or translation research, and we always talked about the phase 1, right? So, and there is a phase 2, there is a phase 3. It's, as I said already, we don't do those phase 2 and phase 3 in my own because I'm a researcher. I'll end up in phase 1, and then if that is working, that phase 2 will be conducted completely by the clinicians and the medical oncologists completely. That is not my work. But what I'm saying is that taking into phase 1, making sure that phase 1 works is a key important point as in, biologists, cancer biologists, therapists, or preclinical developmental. Like, you have to know all those parameters before it goes to the next level, because there are a lot of patient involved, a lot of other things are, analyzed and differently than that what we do. So your drug or your study should go to phase 1. That is, I think, key important point that you should know. There may be a patient sample, like maximum is 15. They may conduct, maybe it will go to 20 patients, that's all. But when you go to phase 2, there will be more patients. And phase 3 is tons of patients, right? They were going to do the multi-institutional, multi-hospital studies and all those things, right? So those challenges are entirely different than what challenges as a biologist we do and conduct in the laboratory. So when you try to design clinical trial, right? That is not our work, but I'm trying to explain to you that when we designed to the phase 1, we did not design. Our science took us there, right? Our science, but when you are designing the therapeutic experiment, for example, Samara designed the AIM-3 with the therapeutic intention of doing, but probably it may go, it may not go because of survival issue. But when she is designing the AIM-3, but when you are submitting the paper to a high impact paper journal, probably they're going to ask you, because they want to translate the study. They want to show, you have to show the translational relevance to your study. They're going to ask you whether, did you do the chemotherapy? Did you do the immunotherapy with this, blah, 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 blah. Also, but you have to know how to do those things and so to develop a trial for them, right? Trial in the future. So phase 1 trial objective is the safety, dosing, pharmacodynamics. That's all. So that's all they collect the information. And designing those biomarker guided enrollment and all those things, it is the next stage. But after the trial 1, even though in the trial 1 they actually do it a little bit. Okay, so the recent trials combining, like, what we did. Examples I already showed you yesterday and today. Not today, yesterday. I showed you all those 2 trials, a phase 1 trial. And do you know all the story why we added the immunotherapy? Right? You know the story. You know the story, without immunotherapy, we also went for a phase 1 trial. You know that story too. So that story is enough for you to design your kind of experiment you want to do. Okay, that actually a good story for you. So there is no need to go what exactly clinical trial will be conducted, because we are not clinicians to conduct the trial or how they do and what they do and understanding it. Okay. So the successful combination therapy, for example, I was telling you about, okay, this is a Nature paper you should read. This is a very recent paper in 2022, because here they're going to explain to you how to do the drug combination. Okay, they have done 2025 drug combination study, And they screened in breast cancer, lung cancer, and pancreas cancer cell lines. Like, there are like 125 samples. They also studied the synergy. They evaluated on the basis of efficacy and the potency. I think almost like 108,000 drug responses. Okay, and also they have they have identified the biomarkers. They have like in here you see the heat map where they see they show you the IC50 values for 1,275 combinations. They combined 51 breast cancer cell line. They have clusters. They're actually clustering and all this. There are a lot of huge information here. I'm telling you, these combinations are available in this paper. Okay, that's what I'm telling you. This is 22. You were in 25. Just imagine in these 3 years, how many other papers came out, and how many drugs or combinations are studied. You don't have to go anywhere and search for it, for the science. You can actually get, by sitting in here, without a test tube, you can actually do it. If you have a knowledge, idea, how to use this kind of data and analyze and think in your own model, analyze and think in your own lab, somewhere sitting at home or laboratory, okay, believe me, you can actually get it. But it's about your awareness. Yesterday I completely told you, awareness. You have to develop that. Read papers, understand it. There are a lot of literature you have to read, day and night. Get up and start reading and search for new paper every day. So this is the key thing. So that's why otherwise our knowledge will be like, we will not be knowing all these things. What's going on? It's already what you're working on. Probably it's already done like 15 years ago. Probably you're just running because you want to run the lab, or you want to just do the PhD, but you're just doing it because the sake of doing the PhD. You may be missing a lot of information that you needed those information. I think that you should not miss it. You have to. For example, people don't. I know many of them. They don't go and read the Nature paper because you don't understand the Nature language. Okay, and believe me, they don't understand the Nature language. Take the dictionary and read about it. I'm telling you, you will learn a lot from those papers. But nature is nature. Okay, that's why you will not be able to understand. But it's okay. Okay, but read, understand, and otherwise talk to someone and discuss. Okay, you can actually get it. These are all English only, but it's about the language they use. Sometimes probably it is not common words that you're going to use it. You use it differently. You have to develop those skills too. And probably if you're not an English speaker, I was not. I studied in Kannada language up to 10th grade, and I went to PUC and I just started using the dictionary. So that's how I learned a little bit of English at that time. So what I'm saying is that it's important you learn yourself if you want to live in the world, in scientific world. You have to learn English, right? And especially the words they use differently, you have to use it. I think this is the great paper. I think you should do it. And this is the same figure I have taken from the same paper in the Nature paper. Basically, what they are trying to establish here is the synergy between many of these cancers I'm talking about. There are a lot of drugs that already know. You don't have to search for it. I'm telling you, there is synergy. They already know. If you see this figure, you already know. There are a lot of drugs, approved drugs, synergy. They can tell you already what exactly is going on. You don't have to search for it. That is the awareness I'm telling you. There are a lot of studies has been conducted already before you even think about it. Okay, so that's why this kind of papers, there are a lot of tons of papers are out there. Collect those information, collect those studies. One paper is enough for you. One paper you can get another one within yourself. Okay, because the knowledge, how you are thinking in your laboratory is different than what I am thinking in my laboratory. Every human mind is different. The way you think, maybe next person will not be thinking. Okay, your thinking may be different. That means that there is a better or worse, that's secondary. But thinking is always different. Okay, when you read one paper, I'll tell you, 10 people think differently about that paper. But there is a common factor. One is that surviving it, or whatever the conclusion is the same thing. But your thinking is different. Means that there is a 10 paper, but the conclusion is the same thing. But the way you conduct the experiment, 10 people, is entirely different. Okay? So what I'm trying to tell you is that the data is out there. Okay? You may have to start thinking and searching information. Okay? And important, I think, another key lesson here is that integrating the therapeutic approach means that you may be doing, for example, in another laboratory, someone else is doing some other type of therapeutic, probably same drug. Okay, for example, MEK inhibitor, right? In some other laboratory, another person published another MEK inhibitor and published differently. And you have to collect that information. Like trametinib is one drug, another one is MEK inhibitor, another drug from different company, AstraZeneca company, some other company. So there may be tons of paper. You have to collect all this paper. Take the common factor from all this paper. Believe me, you will be amazed to see what's going on here. You'll get a new paper, your own, when you collect all this information. That's the beauty of these drugs, right? Each drug act differently, do differently, even though their target may be the same. Okay. There are a lot of immunotherapy combinations are coming out. Okay, nanotechnology, AI-driven drug discovery. There are a lot of AI-driven drug discovery because it's easy to get the information worldwide and collect. When you type something, you actually get that information, whether it is right or not. That is you have to analyze and you have to dig those information, and you as a scientist, you have to make sure that that's the right way of getting the data out. But information is out there. Okay, there is technology developed. Okay, model has been developed. Nanotechnology is also another way. But in nanotechnology, when it comes to nanotechnology, you need to know that why you want to do the nanotechnology. Not because someone else is doing, not because it's a pioneer, not because if the technology is a great technology. No, it's about your scientific problem. For your scientific problem, you have to develop the nanotechnology. Okay, what is your scientific problem? You have to first find out before you do the nanotechnology. Okay, for example, you want to target MDSCs. Okay, you have to target the macrophages, and you don't have the drug directly to target the macrophages, and you want to do the nanotechnology, specifically bind some factor that will go only to the macrophages and hit the macrophages, and then you can actually kill the macrophages and do something, activating some signaling there in the macrophages, then you study. So if your problem, scientific problem, is macrophage related, that's what I'm trying to tell you. So your science has to take you to nanotechnology. Nanotechnology now should not come to you. Okay? So you have to bring the problem first and then think about what technology you want to use it. Okay? So immunotherapy combination, I already told you example, our own example, why we did the immunotherapy. Again and again, as I'm telling you, it's the same thing though. It doesn't matter which cancer you're going to take, you end up doing the immunotherapy. That's why this common factor, I keep telling, it's a common. It doesn't matter which you're going to work on, you're going to do the immunotherapy. So if you cannot do the immunotherapy, it's okay. It's about your work, where it is going is a key factor. Don't worry about immunotherapy if you don't want to do it, or if your paper is not impactful. It's okay if you want to just do the normal thing, it's okay. But do it right. Do it right, validate right, and make sure that is actually reproducible. I think that's the key. Otherwise, you'll not able to sleep later on. Okay. Overall, I think recapping all this work is that integrating approach, molecular mechanism to a clinical trial. So combination, biomarker study. If you think the translational approach is essential for your therapy, but you have to really, the way that you conduct the experiment from the beginning has to be different. Okay? If you want to take your study to a translational, I'm telling you from last 2 days, the way that we have approached, because we were not approaching our science to a translation. We did not know when we did the first experiment, we're going to do the translational. We did not know. But it just took us there. But later, we planned such a way that if we are going to do the translational, and we failed also in one of the experiments, but after that, we went back and figured it out. But important, I think, is that even though we failed, we published in a good paper, but it's not enough, right? The way you start your experiment, for thinking what you want to do in the later on, I think the key factor, right? And your impact factor is 5 or 10 or 20 or 23 or 25 or 50. You have to decide how much work you want to do. Okay, 5, how much work you need, and 50, how much work you need. You have to think differently. Okay, how much collaboration you have to do. Okay, if you want to do collaborative research, then the impact is going to be higher. So that means that you have to go through all these problems and figure it out together, right? So like that, it's about what you want to do, right? How you want to do, where you want to do, and where you want to go from this paper or the work, right? It's about that. So that's the story. Just gave you a wrap up of all the work that we are actually conducting, and also discussed last 2 days. I think if you have any questions, I would like to answer. Any questions?
Hello.
Any questions from the morning? Any questions from today, any day, any talks? Anybody has any doubt on anything? So, who is actually conducting experiment right now in the laboratory? Who is actually doing some experiments in the laboratory? No one? Are all scared? Okay, so you're doing the PhD. So that's why you're doing the experiments. Okay. How many of you are doing PhD? So that's many. So that's good. So I didn't know that this many. I knew that there are many. But that means that definitely is helpful for you. So what type of research you do? I cannot ask you, is it cancer biology or something else? Who is doing cancer biology? Cancer related. Antioxidant is a plant extract study. So is it something natural compound? Some other than cancer, how many? Okay. That's good to know. So you're from food technology, you do food technology. Who else is food technology? Food related. Good. So see, even though you food technology, let me tell you, I was from food technology too. So I'm from food biochemistry. I did a PhD in DFRL. So when I started, I was actually screening the rations for army. Okay, rations, biochemical analysis of rations, and biochemical analysis of all this army, that Silcher and all those people, like freezing condition, non-freezing condition, regular temperature. What is the difference, and what the biochemical assay. We did publish a couple of papers during that time. So also I was running all the GC, mass spec analysis, HPLC analysis, or all those things. So for the food only, it's a biochemistry. Then my PhD was actually on the aging. That means the food aging, right? Meat aging, and because we want to increase the aging, we want to decrease the aging so that we can actually eat it even longer for the army purpose, right? It was important for the army. So I was actually studying to decrease the aging so that we can actually keep the meat for a long time. So, but I've done the biology of it. I studied the biology of it because I want to figure it out. How do I do that? For that, I needed science within. I can put the spices. I can increase, stability. I can do a lot of spices, Indian spices, and mix and match and all those things. And then I think that you can actually store it for a longer time. But that's what exactly we do at home, right? We mix with spices and marinate, and then much better. So, but we can do that. But important, I think, in biology, how do I reverse the aging? That was actually interesting to me. So that's what I did, and that is my PhD program. But we did finally a little bit of that. But what I'm saying. So, but I learned a lot in biology wise. But I learned how do I change it. But it may be probably helping some army, someone somewhere. But how they have used that technology somewhere, something for something, it's a great thing. But good relief, good feeling, right? Because it doesn't matter what research you do, but end of the day, if you do something interesting and conduct something interesting, which actually gives you values to something, It was actually a great feeling. But my interest was some of the factors I studied during my PhD program, during the food biochemistry analysis, whatever, and whenever I was doing PhD program, those factors, the enzymes and the degrades those actin, nebulin, and anything, all those structural proteins was cathepsins and calpains and proteases and all those things. But those are important in cancer also, and during that time. That is the reason that I developed the cancer biology during that time. Means that I always wanted to do cancer biology somehow. But in India, I think at that time there was very less cancer biology, investigators around at that time. So I was actually searching for cancer biology. I went there to US to do the cancer biology. That's all, nothing else. So I joined the aging lab to initially to understand whatever I did, the aging related, that whatever the even a small protein, I wanted to know, because the lady that I joined, she's a space DARPA project she used to do for the defense project. It's actually about aging again. When they, astronauts go and come back, then she was analyzing all those blood samples and all those, biology, and seeing what happens to those factors and all. So, but for me, that was like an exciting factor, because I did the same thing in a smaller level, but I wanted to study and understand how she is actually conducting experiments. I went there and I did for 6 months or 7 months. Then I realized that that is not me, because that I cannot go beyond something in that field. Then I went back to cancer biology, because that is my passion. So when I went to cancer biology, I was like relieved completely, because it is a passion that you believe in something that you wanted to do always. So I went there. But still, when it comes to aging or aging studies and all this, I get excited, because even though I open some paper, and if anything like aging related, I always go back and look at it and see what's going on. That's what you do. So it doesn't matter what you do, but at the end of the day, you always go back and look at it. And I think that's important. It doesn't matter what protein you're studying, whether you're doing about botany or zoology or genetics or food technology, food science, processing or not, the passion that you develop, right? The studies that you're conducting in your laboratory, it has to be something novel, has to be something new, has to be something that you are the one, the pioneer. Okay, you are the one that conducted and investigated. No one in the world has done that type of experiment. Even though you sit in that corner, doesn't matter where you sit, Davangere or Shivamogga or Mysore or Bangalore or Tumkur, whatever, wherever. Okay, wherever you sit there in that corner is important, that what you do, right? That's why this literature nowadays is so good that anybody can get any information anywhere. It was like, we were not able to get even the one paper to read. So I used to go to CFT, DFRL people, the defense laboratory people, can go to CFTRI and use the library. That's the advantage what we had. So I went to CFTRI every day in the evening after 5 o'clock or 6 o'clock, whenever, because the defense laboratory closes 5 o'clock. They only work from 9 to 5. When then the director changed, and my boss became a director, and then things have changed, because I can stay in the laboratory and I can work more because my boss was a director. But before that it was difficult, right? And then during that time, after 5, when they close it, I used to go to CFTRI and read all this related, because it's all food related only. So all these papers and everything was available in CFTRI at that time. So I used to go use the library every day. So I think it's important. You have to search for the information. One another story I want to tell, I always told Joshi. Joshi knows I did not do my experiment, a lot of experiments at DFRL at all. Okay, my experiments, I only was running the gel, and only the photographer Raju was taking the photographs, and I had to go and develop my photos in some other, in a studio in Mysore. I used to go to Gandhi Square, around that area. There's one, photo studio, and then I used to come back and I request Raju again every other week, and I was like, how difficult it is in the university. There's one photographer, and has to come and take a photo of your gel, and it's embarrassing to him, and it's like, for me it is life. So I don't know how to take a photo. There was no camera, there was nothing, that we don't know how to develop all those things during that time. And I used to, to run all these things. Remember, these are all structural proteins. So I had to run a gel like big gel, and the proteins, there are tons of proteins. I don't know which protein is what. I had to find something new protein that no one has seen. That is what my intention is, because I was doing the aging work. So then another one, I used the microscope in zoology in Mysore, and I used to go and use the microscope because there was no microscopy in DFRL that I can use. And then I developed the mice, the antibody for actually isolated the nebulin protein from the gel and then crushed and isolated and all those. And I used to inject in the Mysore University Biochemistry department to the rabbit because I was not able to use it in my DFRL. And inject a rabbit and take the blood and isolate all these protein and things like that. And then when I wanted to do the electron microscope, because it's a structural protein, I had to go to NIMANS, and there is a electron microscopy there. I used to go to NIMANS every other 2-3 months once, and I used to stay there, my uncle's house, and then I used to go there and conduct those experiments, because I had to collect the meat, I had to buy a meat and age them, and we put it in the different fridge, and all those things. And then take the samples to them and cut it and then do analysis and all those. And some of the experiments, I used the friends from ISC and laboratory there, and my things like that. There are many different university institutes I used to run for all those experiments. That's why it came out beautifully, right? When you get a poster award, first award, like oh my God, it's mine. I don't know, like unimaginable, because they see it. When they see it, the effort that you put in, you can actually show up, right? So it's, I think, important. I think the work has to take you, right? You have to have the interest. If you don't have that interest, it's very difficult for you to, long run, publish or do anything. Today, if you see why I was able to accomplish any of these things, because I have passion myself. It's only interest. It's not, my boss was not telling me, my mentor was not telling me to do this experiment. I used to conduct, and I used to go and show, this is what is the changes, this is the paper says this, and this is what I got, and I don't know what to do next, and I'm trying to establish. Then they will give, hints somewhere here and there, mentors, but they may not know the details of it, but the experience they'll give you a hint that somewhere this is how probably is working, right? So that itself is huge. So I think why I ask you different, this thing doesn't matter which one you're working on. You all can become a cancer biologist. You can all become something else. All become a food technology if you want to. If you all like food. So it's about that. It's about your passion. Okay, you can actually develop. Many investigators from cancer biology, they are from chemistry. They are from engineer department. There are many investigators, they are engineers. And I told you one of the example is the sonic, technology that I use for the collagen. She's completely engineer. She changed all the technology to a cancer biology now, and she is doing great. She has got a lot of grants and things like that. Because technology, if you learn something that you can adopt to some other biology, and you can use it. So, for example, if you learn nanotechnology, if you know how to do nanotechnology, believe me, there are a lot of investigators you can collaborate and do those things. You'll be a pioneer in one day, in a couple of days. Like that, there are my friend in Vemula, in Indian Institute of Science, is actually a chemist. He was in Harvard, and he came back to India, became faculty. So he actually designed one of the drug that I'm going to tell you tomorrow. He designed the drug, natural drug, synthetic drug. Believe me, he is expert in chemistry. He has even companies in Boston, and is that expert. Means that even though you're sitting here, your company is running in Boston. Can you imagine? Like that. So if you're expert in something, believe me, you can actually do greater things, in your life. It's about, that's why I was telling you in my first talk that you had to be expert in something. You have to find something that you're interested in. Your mind says you are this, and your body says, no, don't do that, do this. You better do this, because probably you become expert one day, right? Probably that itself is more than enough. You don't have to do much, right? So So this is a question and answer discussion question. If you want to ask anything, please go ahead. We're going to do the one tutorial that is, I think, the targeting cellular signaling and tumor microenvironment. I think that's the morning session.
Yes, sir.
Yeah, go ahead.
Sir.
Yeah.
Yeah. Actually, you answer my question because I want to ask you why you don't go to the phase 2. Okay? But my question, you got a good result from phase 1 result. What you will do to convince the investors to proceed in the next level? As you say that because you are just researcher and you need the clinician, for example. What you will do to convince other investors or the funders to proceed in your result to your next level of phase 2?
So you're saying if you don't do the experiment in the test tube, like how do you convince your clinicians?
Yeah, I mean that you stop in the phase 1
Trial.
right. What you will do to go to convince the investors or the funders to go on the
Go to the phase 2, right?
Yeah.
About your toxicity, your drug combination has to have the less toxicity and also survival, increase in the survival in a cancer patient, right? You will have in the phase 1, you will have like 15 to 20 patients, right? So in the 15, 20 patients, you need to have a good statistics of these patients that these patients survived better than like how you actually showed in the published paper. Like, you survived better, right? At least you should show similar results or approximately that your patients are survived. For example, your patients were surviving before 5 months. At least if you can show like, these patients have survived like 6 months, 7 months, 8 months, 9 months. That is one factor. Another factor is toxicity. You have to have a less toxicity, right? We're going to talk a little bit about that tomorrow, a little bit about it, and then how that toxicity is important, like how to take care of the toxicity if that is something. If anybody is interested to work on, that's a great area too. So just the toxicity, you don't have to do anything. Take care of the toxicity, okay? Improve the patient care. That itself is a huge thing, okay? Yeah. Okay, from morning session, right? So we have this, is it a tea time? I didn't know. Is it? Is it? Okay. Okay. Okay, I think we'll take a 10-15 minutes break, and then we'll come back again, and we're going to go through the tutorial questions. I think I want you all ask me if you have any doubt. Doesn't matter what tutorial material that we are going through. If you have questions, it's fine. If there may be more specific here in the tutorial, if you have any general question, is also fine. Anything you think about in life and whatever you're working on, it doesn't matter plant or the extract or anything like that. It's okay. It's curious, interesting. So anything that you want to discuss and open floor, okay? So it's okay. Don't worry about the material only, whether you understood or not. At least you have the material. If anything that comes in, that you can ask questions. But I think it's also important that during that session, open up. It's okay. Okay. So thank you. tutorial questions, and if you have any questions, you can ask me. What I want to discuss, the same thing what I presented in the morning and also afternoon. I just give you a glance of all those and little bit here and there. I want to tell you the story of some of these questions, and then I also answer to those questions, like, a story again, you know what I'm saying? Not to seriously work on the questions and answer. At least you don't have to worry too much about it. If you don't understand, it's fine. But if you understood the concept of that talk in the morning, definitely these questions are answerable in your own way. But if you don't have it, it's fine too, because that is the story I wanted to deliver and give you, so that you all understand what it takes a PhD students to, accomplish something, and also establish herself to become a great postdoc in a great university and a mentor, and also learn. Even though she was a cancer biologist and she learned immunology, she passioned immunology, and she started working on immunology, right? It's about that. You change as a food biochemist and become a cancer biologist, right? You may be someone like can become something else someday. So it's about what you are interested in, and during your process, while studying, understanding what you are working on, probably you'll learn something and you take that and move on, right? Whatever that you are passionate. Okay. So question is, why is targeting cellular signaling critical in developing therapeutic strategies for PDAC and pancreatic cancer? It doesn't matter pancreatic or any other cancer. So if you cellular signaling, right? So it is critical, because doesn't matter which cancer you want to treat, all these cellular signaling pathways are the one actually helping all these other signaling in other populations, right? Either tumor intrinsic or tumor extrinsic, they all regulate through cellular signaling. Even though you take the tumor extrinsic other cell population, it's also in the air. There is another signaling pathways they regulate. So targeting these pathways, well known in the field many years, that targeting some of these factors. For example, you say one cytokine like IL-1, you can actually block IL-1 too, okay? Like that, you can actually block some cytokine. He was talking about IF-1 alpha. You can block IF-1 alpha, and actually you study, and you get the same kind of targeted therapeutic. But signaling is important. Okay, you have to understand the mechanism. So that's why signaling is important. It doesn't matter what you treat, but as a biologist, you need to tell the story of how did you do, why did you do, and all those things, right? The next is, what role does P38 MAP kinase play in pancreatic cancer, particularly in relation to IL-1 alpha? See, the P38 MAP kinase is overexpressed in KRAS mediated pancreatic cancer, and also it actually regulates, I presented that data, regulates IL-1 phosphorylates a lot of other SP1, other P65, that's a NF-kappa B factors that led to IL-1, lot of IL-1 production. This activates on inflammatory CAV and also it supports the immunosuppressive microenvironment. So those answers that I was giving you is all derived by experimentally conducted experiments and kind of worked on and showing that this has happened. So you basically conduct experiments to prove something, right? To prove something that you have a question. So that's exactly what happened, the overall, right? So there's a question, how does IL-1 contribute to the tumor microenvironment? I don't want to say IL-1 only. Any cytokines you take, chemokine you take, or any factors that you take released from the cell. See, there are many factors released from the cell. Okay, they are all in the bloodstream too. Okay, there are many things released in the bloodstream. Blood is not, it's not blood. It has everything you think of, because it is flowing, going from one body part to another body part in the whole body. That's why blood has a lot of chemical in it. Okay, so in that we were just taking example of one IL-1, Like that, there are so many factors that are running around. Okay, they go one body to another body, one organ to another organ. So, for example, let me tell you, if IL-1 is bad, okay, the bad IL-1 from a pancreas, okay, is released, and if it is really bad, it is actually flowing to other organs too. But it's only the factor is running around. If the factor is going and running around, it is not going to make the metastasis. Your cell has to go, then only it will be metastatic. Okay, your factors may be in your body, bad one, good one, are all there running around, but they are okay. Okay, there is nothing wrong. But that's how the bloodstream work. They go to heart also, they go lung, they go everywhere. But the bad part is that your cancer cell, which actually metastasized to another organ, is a bad thing. You have to control that cancer cells to go out of your cell type. That's why if you can reverse, like for example, I always say, in aging, I wanted to reverse the aging. The same way, I want to reverse the cell shape towards the normal, how much ever I can. Every day I think about it, like, how do I do it? Probably one day we may do it, but what I'm saying is every day we think about that one issue that I had it like in 25 years ago. I keep thinking, how do I reverse it? What actually makes? You keep reading and probably one day you will figure it out. Probably not. It's okay, but you get the knowledge. At least I read a lot of papers. You know what I'm saying? So like that, the factors, we are only giving you example. But you have to understand from that one example, like so many things that you can actually study, like, understand also, okay? What experimental evidence supports the use of P38 inhibitors in reducing IL-1 expression? See, we have showed the preclinical studies, pharmacological inhibitor like, MAP kinase inhibitor, and also we studied with the knocking out the P38 is important. You don't do just only the chemical. You have to do with the knockouts and gene regulation, knocking down and, all those experiments and prove your hypothesis in, genetically and also pharmacologically, always, okay? Which transcription factors are implicated in the P38 MAP mediated regulation IL-1 and how are they affected by P38 inhibition? The transcription factor, I already told you, SP1, NF-kappa B, which are critical for IL-1 transcription. The P38 MAP kinase inhibition reduced this phosphorylation, nuclear localization. You saw all those things, and also it led to a reduce the IL-1 promoter, also IL-1 expression. So what I'm saying is that when you study, something pop up and in your study, and you need to understand either cellular intrinsic, extrinsic, and also nuclear, cytoplasmic, and if it is nuclear, what it is, and cytoplasmic, what it is, and whatever the molecule you are studying, whether it is a cytoplasmic, it's related, or the nuclear, you have to work on and study. You have to experimentally prove that though, cytoplasmic and also nuclear, then divide your experiment and conduct the way she conducted Samara, like, herself. Like, then she did the inhibitor study and also she did the knockout study. These kind of things probably give you a lot of answer. So how does the inhibition of P38 affect the CAF activation in cancer? So by reducing this IL-1 alpha P38 inhibition, it actually, you saw, it actually impairs the inflammatory activation of the CAFs. So also you saw how this growth factor cytokines, pro-inflammatory cytokines, and growth factors of CAPS, which can also reduce the desmoplastic reaction. She actually showed some fibroblastic, reducing the fibroblast cells. Like in IHC, she did staining the trichrome blue and Sirius red and all those things. And she showed like that. In each cancer, it's important is that what is that important in each cancer? That in our pancreatic cancer, stroma is important to study. So that's why she did that. In other cancer, there may be something else. You have to do those and conduct those experiments, right? So what are the main differences between IL-1 alpha, beta? You already know. IL-1 alpha, tumor intrinsic, epithelial, you already saw the data and everything. And she did IL-1 beta in the other population, macrophage population. She saw IL-1 beta was there. And then she inhibited the P38 in IL-1 in those population, and she saw that IL-1 beta went down. So basically, you saw the difference, that she did not do IL-1 beta in the tumor intrinsic. She only did with the other population. So like that, if someone has published some paper on, okay, what are these do, what are these going to do, that is true. What she saw, what others have published in paper is also the same thing. So she just proved it in our paper too. That's important. Proving the idea and the concept ideas from others, again in your work, is the important thing. So you have to prove it and keep going, proving it, and also proving the hypothesis that you generated, right? So describe the impact of P38 inhibition on the tumor microenvironment. So it's just when you inhibit P38, it's just not only reducing the inflammatory CAF activation, right? It also remodels the immune components. She studied each population. She studied like macrophages, monocytes, MDSCs and T cells component. She saw all the T cells, CD4, CD8 went up. You know what I'm saying? So like that, she had to study different cell component and do. That's the beauty of the technology that we have today on single cell sequencing. So how does combining MAP kinase inhibitor and chemotherapy improve the therapeutic outcome in PDAC? Because the chemotherapy is a standard therapy already improving in the patient, right? So now you add with the synergistic combination of this P38. So it did work. It did increase the survival. It is not very great, but I think you did good job. Like in at least 2 to 3 weeks is a huge thing in pancreatic cancer. So that's a good sign. So what experimental methods are used to assess the effects of P38 inhibition on IL-1 transcription? If I ask you, you might know already, you might not know. There is a lot of chromatin immunoprecipitation, CHIP assay she conducted to measure the transcription factor, like binding to IL-1 promoter. That's exactly what she did. You have to do the same way. You have to conduct those kind of experiments to prove your scientific investigation. If you say something is regulated, that means that you prove it. Okay, you cannot just say, oh, it's regulated, I move on. No, you have to prove it. So that's what she did. So she proved it herself again and again, right? So it's a key thing. What is the significance of IL-1 receptor localization in CAFs? You already told you, IL-1 receptor only present. She showed you in all the single cell sequencing data, it was present only on the CAFs, nowhere else. So that's why she was very confident that her IL-1 alpha has to go and bind on the CAFs. So because the receptor is right there. So she showed that IL-1 receptor is expressed there. So things like that. So next question is what ways does P38 MAP kinase inhibition alter the inflammatory and stromal phenotype? So the P38 MAP kinase inhibition reduced the inflammatory markers. She showed the IL-6 also. These are inflammatory markers. And CXCL1, I told you we had a paper in the cancer discovery on CXCL1, and it's a great marker too. It's a pro-inflammatory marker. That is also reduced. And she showed a lot of other phosphorylated CAFs because STAT3, because STAT3 is also inflammatory marker, and she showed all those things. So that's a great combination of all these markers, tumor promoting markers. She showed with P38 inhibition. Again, all these markers went down. So next question is how does the CAF specific deletion P38 MAP kinase influence tumor growth in PDAC? So see, the CAF specific deletion in P38 she conducted because the P38 is also expressed in CAF a little bit. That's why she did. And it has been shown reducing the tumor weight and the fibrosis. And the overall, when you do the drug study, when you use the drug, basically the drug is going everywhere, just not the pancreas. It goes every other organ, okay? Every population, like for example, immune population too. That's why the drug can hit everywhere, right? So basically, it did work. It did work because there was P38 expressed in the calf. Probably the drug went and hit the calves also. That's why the overall the therapy worked. Okay. So what future therapeutic implication can be drawn from this targeting P38? See, targeting this P38, it opens up a lot of avenues for us. So we can do like personalized therapy. Particularly with the patient with IL-1 alpha, IL-1 beta. If we can see in the cytokine in the blood sample, if they have a high IL-1 alpha, that means that you can expect probably high P38. So you know what I'm saying? Prediction. It's just we don't know. It's just a prediction. So it also provides the strategy to combine with the immunotherapy. We saw that we can actually detect all this in the cells. Some of the cytokines can also activate on the immune cells. So kind of is all prediction. Probably in the future, with the technology changing in technology, we can actually use like personalize something, can do much better work than what we did. So how do the findings on P38 mediated signaling support the design of the combination therapy? See, the highlight that we did on disrupting the key signaling pathway P38 can simultaneously impair this cell-driven inflammation, modulate tumor microenvironment. So, but overall the dual action of this P38 worked when we used the P38 drug because it killed everywhere. So, I mean, basically there are many questions are there. Any research you take, there is a lot of questions arise even after the work is done. Like now, did you take care of the resistance of the mechanism? Did you check the resistance mechanism? Did you check that, this, that? But you cannot answer in one paper, one publication, everything, right? You saw how much work that has been done, and you will not be able to answer all of them. But there is a lot of questions, but we can actually answer one by one later on in the future. But that's how it is. That's how the research is. So, any questions? Anybody?
So did you see any changes in P53 levels after targeting P38 because it activates P53 also? So
correct, but no, we did not check the P53, right? Yeah, P53. But remember, our model has a P53 mutation already, right? So the one of the model, not the other model without the P53. Now we don't profile the genomic profiling, but when they conducting the patient surgery, after the surgery, if they are treating this drug with to the patient, what they do that after surgery they can get the samples like tissue samples and the blood. They actually take the tissues and also do the genomic analysis. So they do check actually whether what happened. So.
And also, sir, have you checked any upstream genes after targeting P38 and MAP kinase pathway? Any upstream targets have you checked? What is the?
Yeah, you can actually check, but we kind of derived already. So people have already published that it is a KRAS mediated activation of P38. She actually also showed in her paper KRAS mediated activation. Above the KRAS, already we talked about EGFR and SARC and all. There are other signaling comes. So we don't want to dilute the work because it's a lot of work to show everything in one paper. But she actually did all those screening before she went into see the activation of IL-1 regulation and all this. So, any questions? Any general question? Anything about life? Now go back and turn it back because it was fine, we just had to hold it.
One is related to metastasis. Is there any stage, sir, in cancer like metastasis starts? Like we have different stages of cancer. At which stage the metastasis starts? And what makes it to start metastasizing?
So it is not that what stage it starts. It starts after when it is tumor completely. So it has to be a full-grown tumor. The metastatic cells can, after that start metastasizing. And also sometimes in some of the tumor, cancers we have seen even early on, a little bit earlier than the, during the tumor stage. Okay, when, for example, in PANIN, we talked about PANIN 1, PANIN 2, PANIN 3. The PANIN 3 is the tumor. So it can start with the PANIN 2 and PANIN 3 any time. These cells can change the shape. And once they change the shape, they start going to the bloodstream. Through the bloodstream, it should be able to enter the bloodstream. So it can be anywhere there. But it cannot be early on because the shell shape is different. So it's hard for them to go and metastasize. Okay, it all depends on the shell shape, right? That only changes during that transition to the cancer. Okay.
And my second question is importance of metabolomics in cancer research.
So importance of metabolomics,
Yes,
right?
Like targeting any metabolic pathways will help us to.
Very important, right? See, when you say cancer, the metabolic pathways are high. Sugar, so sugar, they love sugar. The cancer cells love sugar. They want sugar. They want energy. So that's why mitochondria release a lot of energy, a lot of ATP. And glutamate is a bad thing. The high glutamate that is what they want. So if you are giving the cancer cells what they want, I think they're gonna grow like crazy, right? You have to reduce the sugar. You have to reduce the energy from the mitochondria. See what you can do. And also glutamate you have to reduce. And see what you're eating, what you're drinking. That's why doctors say you have to do this. Don't eat that. You know what I'm saying? The reason is that you have to control some of the food that actually giving them the energy, right? So you control those things, and I think can be controlled. So it's about that. So metabolism is a key factor. So any amino acid, all this glutamate go through the metabolic pathway, and energy is to the metabolic pathway. So that the whole metabolism is a highly important pathway to study. Okay, yeah.
Sir, is it possible to stop like metabolic pathway in tumor tissues?
Yeah, there are a lot of studies. Remember, cancer cells are more brainier than the normal cells. Means that they find a way to survive, right? So they find the way to survive. So it's just that. So they find another pathway to survive in it with another one pathway. That's how the cancer cells are. But they will always find, there is a lot of targets, there are a lot of studies have been conducted. They are conducting, metabolic pathway is one of the important signaling right now in the cancer field. Thank you. Any question? We have another 5 minutes. You have to speak louder.
Sir, I have a question. I wanted to know how did you get the DRDO fellowship for your PhD?
How many?
How did you get the DRDO fellowship for your PhD studies?
Or DRDO fellowship. So DRDO, they don't have like CSIR at that time. I don't know 25 years or what they have done. I'm telling you the story 25 years before. Okay. So during that time, DRDO announced, mine was the first batch DRDO fellow. Means that was the first year that they announced the DRDO fellowship. I was the first batch student, got a JRF. After that, they keep announcing every year JRF fellowship from DRDO. I don't know what they're doing now, but I'm sure they're doing the DRDO fellowship. That is not like an exam. They actually announce the fellowship, you apply, and they interview only selected candidates in those institutions, And then after the interview, we had a written test, and they also did not know what is GRS fellowship at that time. You know what I'm saying? Because there's a defense, first time students coming, like, they don't know what the DRDO fellowship means. You know what I'm saying? That stage I'm talking about. But we had the entrance exam, and after that, after the entrance exam, we had the interview. Morning entrance exam, afternoon interview. After the interview, they've selected. Only they selected, I think, 4 out of one institution. They only selected 4. So like that. But there was, I don't know now. If anybody has any information, let me know. But I don't know now what they are doing. This is 25 years ago. Maybe 27 years ago. Okay? Good. Okay, thank you so much. If you have any other questions, so we can talk later outside. So thank you so much. I think we had a great day. Hope you all enjoyed it. I think tomorrow is a short day. So, pardon? Your tutorial too. You want to go through the tutorial too? Can I ask you all the questions? Huh? So, what question do you have? What questions is bothering you? Yeah, tell me what question is bothering you.
Sir, you are mainly working on the signaling pathways, isn't it? But there is another strategy against cancer is inhibition of certain enzymes.
Inhibition of?
Enzymes which are involved in, for example, cell replication like telomerase, which stops the cell replication so that we can stop the proliferation of cancer. So what about the work or that strategy of targeting certain enzymes which are very necessary for the cancer cell proliferation and even in metastasis also?
Yeah, you can target anything. You can target enzymes, you can target products from the enzyme, substrates you can target. You can target whichever is important to kill cancer cell and stop cancer cells growing. Anything that is enzymes also same way.
Like some are like inhibitors they are working on, like
so
that inhibit particular what you call enzymes which are involved in cell replication, so that we can stop the replication.
Sure.
Many of them are using, I think they are used as
anything,
chemotherapy.
Anything novel idea you have,
Yes.
or people are working on a lot of enzyme studies too. But anything novel ideas which actually kills cancer cells is important. It doesn't matter which one, right? So anybody wants to know anything from the other tutorial? I think there are a lot of questions. Or if you have any questions, tell me tomorrow.
Sir, one. How are therapeutic agents typically identified? How can we identify a target?
How can you identify?
A therapeutic target in case of cancer therapy.
Okay. Means that, like for example, SARC. That's what I've given you a lot of information. I told you a lot of websites to go through and a lot of softwares to analyze. And the day that when we started the SARC story, okay, I told you the SARC story. How did I do the SARC, right? I told you about who was working on SARC in my Vanderbilt University, and why did I start SARC, right? I didn't know which one to start with. At that time, there was not many signaling pathways, because in the pancreatic cancer, I had to start somewhere. So I was actually searching all the molecules, which one to start with, right? Then the SARC popped up. The reason is that whether the SARC is highly expressed or not, I didn't know. Okay, so that is the first thing that I did. I looked at the SARC, whether it expressed high or not, because I knew there is a neighbor who is a SARC person. But if anything happens to SARC here because those are different time. Today I may not do the same way, right? Because I have a lot of websites. I can analyze a lot of things. I can correlate with my other studies, and I can come up with a lot of ideas. But at that time there was not these information were not there. So this is what I did. I started with a neighbor, who actually had a SARC antibody also, SARC knockout studies I can do, overexpression studies. That is exactly the tools that you need whenever you start anything research, right? You need a tool. But good thing is that SARC was expressed high in pancreatic cancer, in tumor, when compared to normal. So I was fine. At least I thought I will get a small paper out of it, whether it works or not, right? That's how I started. Look where I went into.
Now if we want to start now, for example, we are starting now.
I gave you a lot of
How we can
information.
identify targets?
I've given you a lot of websites to look into, study. I'm not telling you what you should do. You already got everything that, what do you need to do? Okay, you better start working, and so that you have another day if you want to clarify any of your doubt, right? So it's about you. Okay, we only tell you information. I cannot tell you target. I cannot tell you where to start, how you start, but I can give you my story. Okay, you have to think about you, your situation, where you are and at right now, and what you want to do. Okay, you have to figure it out. Okay, you have to search for it. I only tell you where the information is, but you have to search for it. Get the doubt yourself that whether you're searching or not. You have to doubt about yourself every step of your research. Then only you can get an answer. So don't say that you got an awesome data. Immediately you may say that may be not right. Okay, if you come up with awesome data, if you come to me, then I'll ask another question. Where is the other one? Then you may not be happy. Things like that. So it happens. It's common. So disappointment is very common. So you have to work hard. Then only you can achieve. Okay? So if you think that all the information will be given out to you, that means that you will not be able to achieve, because that is not yours. If it is, if the information, whatever the science is, want to be yours, you have to work hard. You have to figure it out. You have to find out. Okay? So the story of the SARC, story of the P38, how she started. I told you in the morning how she started. That story is enough for you to dig the story. Like, for example, neighbors and the laboratory, what you worked on. If you are not in the pioneer lab, then find one. Join if you are interested in research. Okay? Then you find a good paper. You may publish another paper, right? But if you want to do yourself, it's fine. You have to work hard. More and more. Find a way. Okay? See, 15 years ago and today literature is changed. I just gave you 2022-23 papers. Believe me, if I had those papers at that time, probably my science will be different. Probably I would, I don't know whether I will be able to survive all these patients, but what I'm saying will be different for sure. Because there is a huge set of data there. Believe me, I'm also looking at it every other day. So, because I also get doubt of this drug, that drug, and synergy of all those, I also see the same. I'm giving you the same information to you. So it's easy for you to look at it and find out, right? So hopefully you'll publish a good paper now on. Yeah. Okay, thanks. All good? So we can wind up today. Okay, if you have any more questions from the other tutorial too, please ask me. Okay, tomorrow. So we will complete today. So you have to come, right? Okay. Thank you, everyone.
Good evening, all. I thank Dr. Nagaraj Nagatihalli to have presented a wonderful his works here, and I thank all the participants to have participated and have been with us till the day 3, and I thank you all. Thank you.